Welcome to BrainSTIM 2020. I'd like to welcome you to the 6th annual Brain Stimulation and Imaging meeting. Our first meeting was in Honolulu in 2015. And at the time, I realized that there was a need to bring together people doing brain stimulation with those doing neuroimaging, that we need collaboration as a part of what we do. And we need knowledge to transfer between people doing brain stimulation and people doing neuroimaging in a way that benefits both sides. So starting in Honolulu, then Geneva, Vancouver, Singapore, and Rome, this year, we should have been in Aalto, Finland, but instead we're all home waiting for the coronavirus to pass. So I would like to send out a tremendous thank you to Dr. Risto Amalviemi and his collaborators at Aalto University. They've done a tremendous job putting it together this year, and I really appreciate their efforts. And I also wish you a safe and hopefully pleasant time at home. And I hope spending time with BrainSTIM, you'll learn something useful. So what are the burning questions? To me, after spending a few years doing this work, I wonder, how much should brain stimulation and imaging encompass? Should it include every form of energy that we could find? Should it include every form of neuroimaging? And putting all that together, how much benefit can we derive? In the end, I think yes, we need to include as much as we can. If we do that, what are the best methods? How do we tell out of all the possibilities what we should pursue? There's some basic criteria that we should follow. Paramount is safety. If we're putting things into people's bodies, we're putting energy into people's bodies, we want to be sure it's safe. And safety is of utmost importance. Effectiveness is another important issue. Would a certain energy protocol produce a change in the body that's useful? There's so many methods and so many ways to do it. How can we get the most effective? We should probably concern ourselves more, not with most effective, but with effective enough or more effective than currently available. And it's the same with safety. A number of pharmaceuticals are not safe, not entirely safe. If we come up with a method that's equally effective but safer, that could be a reason to pursue it. If we come up with a method that's more effective and safer, that could be a reason to pursue it. We shouldn't expect any method to be entirely safe, but if it's safer than the best method that we currently have available, we need to pursue it. Cost is an important issue as well. If it's too expensive, no one will be able to use it. But if it won't make companies that produce it a profit, it probably won't be developed the way that healthcare is designed currently. There's very few places that don't concern themselves with making money in healthcare. Finally, ease of use. Would somebody who's not a trained neuroscientist or a trained neuroimager be able to follow the protocol that we've put together? We need to design protocols and methods that someone who's intelligent but untrained would be able to follow. And finally, for this year, I send out a tremendous thanks to our sponsors. Even though they couldn't come, they were still able to provide us funding to be able to put this meeting together. So please look up their products and get in touch with them if you can. And thank you very much for attending. And I hope you have a good couple days with BrainSTIM 2020.
Welcome to BrainSTIM 2020, organized at Aalto University in Finland. First, I would like to thank Vince Clark, the series organizer of the BrainSTIM conferences. Vince is professor of psychology and neuroscience at the University of New Mexico and director of the Psychology Clinical Neuroscience Center in the same university. We're going to have a great program, including 3 keynote speakers shown here. Professor Stephanie Jones, Professor Jari Hyttinen, and Professor Vince Clark. The organizing committee is shown here: Erika Haaksiluoto, Satu Jääskeläinen, Mikael Nietzsche, Gianluca Romani, and Ulf Zeeman. I would really like to thank the local organizing committee, which has done most of the work in preparing this remote conference. Aino Tervo, Marianne Rofta, Jaakko Nieminen, and Timo Roine. The local organizing committee posed this question to both Vince and myself. What is the most burning question in neuroscience? Well, you can make different kinds of questions that are big and important, but perhaps not burning immediately. How does the brain work? What is consciousness? And so on. So these, I don't think, are the immediately burning questions. So what is the most burning question in neuroscience? Here's the answer. This question itself. What is the most burning question in neuroscience? I wish you welcome to BrainSTIM 2020.
Audiot. Hello everyone and welcome to the BrainSTIM 2020 studio. My co-host here is Aino Tervo.
And may I introduce my co-host, Jaakko Nieminen. For the next 2 days, we will have great content, many oral talks, and excellent posters. Hope you enjoy the BrainSTIM 2020 with us.
Hello, everybody, and thank you for joining my talk on the impact of functional connectivity of the motor system on the resting motor threshold. My name is Melina Engelhardt, and I'm a PhD student at the Charité Berlin. The resting motor threshold is commonly defined as the smallest stimulation intensity to reliably elicit muscle-evoked potentials in a target muscle. On a functional level, it is defined to measure excitability of the corticospinal tract. RMT is one of the most commonly used measures in studies using transcranial magnetic stimulation. It is often used as an outcome to measure corticospinal excitability, for example, to quantify changes due to a disease or effects of an intervention. It is also used to scale stimulation intensity of other TMS or rTMS protocols, and this goes way beyond the motor domain, for example, extending to therapy approaches in depression. This highlights the need for a very thorough understanding of what the resting motor threshold means, what influences it, and what might confound measures of the RMT. One of these factors that has been identified a while ago already is the coil-to-cortex distance. With an increasing distance between the coil and the cortex, also the resting motor threshold increases. Similarly, with an increasing age, the RMT increases. Other factors that have been suggested include sleep or awakeness and white matter properties. There are many other factors that have been researched in terms of contributing to variability in the resting motor threshold, and most recently, functional connectivity of the motor system. In a paper that was published in 2017 in Brain Stimulation, Dr. Charlotte Rosso identified an impact of functional connectivity on the resting motor threshold. Functional connectivity, briefly said, means a correlation in activation in different brain areas. We can quantify activation in brain areas roughly by measuring the bold signal. We can then extract the bold signal from, for example, the right motor cortex and correlate it with activity in the left motor cortex. If there's a high correlation, it means that both areas have a high functional connectivity. Dr. Rosso found that there was a positive correlation between functional connectivity between M1 and the dorsal premotor cortex and the resting motor threshold. She then suggested a model that explained variability in the resting motor threshold by age, coil-to-cortex distance, functional connectivity between M1 and the dorsal premotor cortex, and cortical gray matter volume. Going on from there, the aim of our study was to validate and replicate these findings on functional connectivity and the resting motor threshold. We also included the coil-to-cortex distance and age in our analysis, similar to the study by Rosso. We extended their model by including data from both hemispheres. Lastly, we also wanted to include the gray matter volume. However, I can't present these results today yet because they're still being analyzed. Our data set consisted from data from 2 studies with a total sample size of 38 subjects with a mean age of 37.5 years and roughly 50% females. Therefore, our data set is roughly double the size of the original publication. Our study protocol consisted of an MRI with an MP range and resting state fMRI sequence and an NTMS motor assessment. The NTMS motor assessment was conducted on a NextStim NBS5 stimulator. We'd identified the hotspot for the first dorsal iterosius muscle, so the muscle for the index finger on the hand on both hemispheres. On these hotspots, we defined the resting motor threshold using the Rossini-Rothwell method. Briefly said, this defines the RMT as lowest stimulation intensity with at least 5 out of 10 positive stimulations. The MRI scans were conducted on a 3 Tesla magnetron trio Siemens scanner with a 32-channel head coil. For analysis of the resting state fMRI functional connectivity, we followed the pre-processing steps used in the paper by Rosso and colleagues using the Toolbox. We computed Roy-to-Roy functional connectivity using the human motor area template as an underlying atlas. This deviates from the protocol used in Dr. Rosso's study, as they used subject-specific ROIs drawn on individual FA maps. We chose our approach because we felt it reflects common usage of ROIs and common use of atlases in fMRI studies, and is a very standardized and objective way to identify ROIs. However, we picked our atlas so it matches the regions used in the original study very well and made sure that our registration of the atlas for individual subjects was matching the structure very well. The regions included in the atlas were primary somatosensory and primary motor cortex, supplementary motor area, supplementary motor area, pre-supplementary motor area, premotor area dorsal, and ventral. We also measured collateral cortex distance on the MPRH sequences, again following the protocol proposed by Rosso and colleagues. For statistic analysis, we followed two approaches. First of all, looking at a subset of the data for only the dominant hemisphere, according to the results proposed by Rosso, and then extending these results by looking at both hemispheres. We computed correlations between the resting motor threshold and the functional connectivity between atlas regions, age, and collateral cortex distance. We did the same analysis using linear mixed models for both hemispheres. Next, we tried to replicate the multiple regression model proposed by Rosso, including age, collateral cortex distance, and functional connectivity between M1 and the dorsal premotor cortex. However, it should be noted that in the paper by Rosso, this includes also the cortical volume of the dominant hemisphere. And finally, for both hemispheres, the same model as a linear mixed model. So let's take a look at the results. I'm not going to go into detail about the results from the correlations because there were no significant correlations except for the ones included in the regression model anyway. The regression model for the dominant hemisphere showed an effect of collateral cortex distance on the resting motor threshold. However, we could not find an effect of functional connectivity. This is supported by a Bayes factor suggesting that the null hypothesis is 5.8 times more likely than the alternative hypothesis. So this suggests that there is not only an effect of collateral cortex distance, but not functional connectivity on the resting motor threshold. Looking at data from both hemispheres, we saw essentially the same results. There was an effect of collateral cortex distance, but not of functional connectivity. And again, the Bayes factor suggests the null hypothesis over the alternative hypothesis by a factor of 8.3. So as you can see in these graphs, there is a nice positive linear relationship between the resting motor threshold and collateral cortex distance. However, no such relationship can be observed for the functional connectivity. So with those results, we then contacted the original authors again and asked them for their opinion on these results, as well as any ideas where our methods could have deviated from their protocol. And one thing that became apparent is that while it is not stated explicitly in the manuscript, Dr. Rosso conducted the MRI scans directly before the NTMS measurement. As I said, for our study, this was not crucial, so some of the MRI scans were conducted right before the TMS scans, some were conducted several days apart. Therefore, we figured timing of the MRI and TMS might explain our different results. And what we did next is we identified the subset of subjects that had the MRI directly before the TMS measurement and looked at their data. However, I should say that those were only 7 subjects, so all of these results should be interpreted with caution because they're highly underpowered. So now again, the results for the dominant hemisphere with only those 7 subjects. We still did not see any effect of functional connectivity on the resting motor threshold. And this is still supported by a Bayes factor favoring the null hypothesis over the alternative hypothesis by 2.38. So again, we see that functional connectivity did not impact the resting motor threshold. Similarly, for both hemispheres, functional connectivity did not impact the resting motor threshold, and a Bayes factor of 2.94 again favored the null hypothesis. So, taken together, we could not replicate findings of impact of functional connectivity, neither between the primary motor and premotor dorsal cortex on the resting motor threshold, no other functional connectivity values between other atlas regions. We could see the well-established effect of collateral cortex distance on the resting motor threshold, and we could see some effects of age on the resting motor threshold. However, they were mixed and not very strong. This might be due to some missing co-founders in our analysis, such as the cortical volume of the hemispheres. Lastly, we also did not see an effect of hemisphere on the resting motor threshold. So now we thought about why we could have different results than the study published by Dr. Rosso. And one possible explanation is that our analysis did not include the cortical volume yet. However, we think this is unlikely to have influenced our regression and mixed models, because also in the correlations there was no relationship between functional connectivity and the resting motor threshold. Next, our TMS system was different than the one that Rosso used. However, both systems were neuronavigated and the range of resting motor threshold variability was roughly the same. Then again, our MRI protocols and scanners were slightly different from the ones that Charlotte Rosso used. However, this is again unlikely to explain the different results, as any effect should be robust against slight changes in MRI protocols. Then, like I previously mentioned, there were some differences in timing between MRI and NTMS. However, looking at the results from our subset, it doesn't seem like this explains different effects either. And lastly, we can look at the different ROIs, which remains a factor of uncertainty regarding the comparability of our results. However, we would argue that our atlas matched the regions selected by Dr. Rosso very well, and we made sure that registration for the atlas onto the subjects was good. Therefore, again, it seems unlikely that this caused the different results, however, it cannot be ruled out completely. So, taken together, we could not see an effect of resting state fMRI functional connectivity on the resting motor threshold. We saw that cortical-cortex distance remains the most important predictor of the resting motor threshold, and that the factor of age remains inconclusive, potentially due to missing confounding variables. And with that, I would like to thank my supervisor, Dr. Thomas Picht, as well as Fabia Roth and Leona Kawelke for contributing data to the study. I would like to thank Professor Carsten Finke and Darko Kumenic for helping with the resting state fMRI analysis, and Dr. Ulrike Krittner for answering any statistical questions I had. I would also like to thank the team of the BCAN for giving us access to the MRI scans and the MRI machines. And I would like to thank the Einstein Center for Neurosciences and the Medical Neuroscience Graduate School for funding my PhD and supporting me throughout. Lastly, I would thank the organizers for this great conference, and thank you all for listening.
Hello, my name is Marta Bortoletto from Brescia, Italy. Although we cannot meet in person this year, I'm glad we can still meet online, and I want to thank the organizers for that. Today, I'm going to talk about the first of a series of studies we are conducting aiming at defining measures of conduction delay of the corpus callosum by means of TMS evoked potentials. And in this study, we have focused on the motor system. The conduction delay is basically the time that it takes for information to travel from one area to another along white matter tracts, like the corpus callosum. The corpus callosum is actually the major tract that connects the two hemispheres, and it has a crucial role in regulating the exchange of information between them. The downside is that the corpus callosum has a long conduction delay, which means that it's a slow connection compared to intrahemispheric connections. For this reason, it has been suggested that the long transcallosal conduction delay might be a factor that led to hemispheric dominance throughout evolution. Basically, a more segregated information processing might have evolved to avoid multiple passes of information across the corpus callosum. However, still the hemispheres need to some exchange of information, so we should still be able to see an effect of transcallosal conduction delay on behavioral performance, mainly when tasks require high timing accuracy. The main problem here is that currently we don't have a very good measure of transcallosal conduction delay. There have been pioneering studies using lateralized effects of reaction times and event-related potentials, like in the Poffenberger paradigm, but these measures do not really provide a measure related to a specific tract. Other studies have used TMS, and they have given a good estimate of transcallosal conduction delay. However, these measures still rely on peripheral output, and they are limited to the motor system. They cannot be extended to other areas of the brain. Therefore, we thought to use the TMS-EEG co-registration approach, and specifically the TMS evoked potential. With this kind of technique, when we stimulate with TMS, we can activate a target region at a specific time, and after this activation, it is possible to record through EEG the secondary neural responses that have been generated to the connected areas. For example, an homologous areas that is connected to the target one via the corpus callosum. And we have tested this technique on the motor system, but the good thing is that if we can validate this technique, we can extend it to other areas and to other systems in the brain. In the motor system, we know that the corpus callosum mediates interhemispheric inhibition with a transcallosal conduction delay of about 10-20 milliseconds, as it has been shown, for example, in ipsilateral silent period paradigms. In this kind of task, the participant is required to activate their muscles ipsilateral to the TMS. When the TMS pulse is given, it is possible to record a temporary reduction in muscle activation. And this phenomenon is thought to be related to signal transmission through the corpus callosum, which inhibit the contralateral primary motor cortex, and eventually this inhibition travels along the corticospinal tract, reaching the muscle. In this kind of paradigm, if we add EEG electrodes on the on the scalp, we can imagine to be able to record this response in the contralateral primary motor cortex by means of TMS-EEG co-registration. So here is our paradigm. We recorded TMS-EEG during the ipsilateral and the ipsilateral silent period in the muscle during 4 conditions of an ipsilateral silent period paradigm. In these 4 conditions, we stimulated either the left primary motor cortex or the right primary motor cortex. In the no-task condition, participants were required to just activate the ipsilateral muscle and be relaxed with the contralateral muscle. In the task condition, the participants still activated the ipsilateral muscle, but they were also performing finger tapping with the contralateral hand because this has been shown to increase the interhemispheric inhibition. We also added DTI measures of the body of the corpus callosum, which is the part of the corpus callosum that connects the 2 primary motor cortices. Here you can see our TMS-EEG data. We were able to record nice TMS evoked potentials in all 4 conditions, but we were specifically interested in an early component occurring perhaps around between 10 and 30 milliseconds. We were looking for a component possibly in the contralateral hemisphere, the hemisphere contralateral to the stimulation, and possibly with a positive potential. That's because positive potentials are associated with inhibition in the motor system. And as you can see, we actually found this component that we called P15 because the peak was around 15 milliseconds as latency, and it was contralateral to the stimulation and positive. And we were able to record it in all 4 conditions of our task. First, we tested if the P15 is a measure of interhemispheric inhibition. For this, we applied linear mixed models with P15 as a predictor of ESP amplitude. And here you can see that we found a positive relationship so that the higher the amplitude of the P15, the higher the ipsilateral silent period area, suggesting that the P15 is a measure of interhemispheric inhibition. Our second aim was to find the relationship with the corpus callosum, and more specifically to see if the P15 latency can be a measure of transcallosal conduction delay. For this, we applied linear mixed model with DTI measures as predictors, so the microstructural integrity of the corpus callosum as predictor, and the P15 latency as a dependent variable. And we found a significant relationship with the axial diffusivity and the P15 latency. Importantly, the axial diffusivity is the diffusivity of the water molecules along the axons of the corpus callosum, which means that the easier the molecules can travel along the corpus callosum here, so the easier the signal can pass through the corpus callosum, the shorter is the P15 latency. So with these 2 evidence, we thought that we can sustain that the P15 is a measure of transcallosal inhibition and its latency is a measure of the conduction delay. Therefore, we use the P15 latency to test the impact of transcallosal conduction delay on behavior. And in this case, we ask participants to perform an in-phase bimanual sequence of finger position movements, and we measure the inter-hand interval, which is the time difference between the taps with the two hands. Well, specifically, a better bimanual coordination is reflected in a smaller inter-hand interval because it means that the tapping is more simultaneous. So our hypothesis was that a short transcallosal conduction delay would lead to a lower inter-hand interval. We tested this relationship separately for the left and for the right M1 stimulation. And as you can see here, on the x-axis, there is the P15 latency, while on the y-axis, there is the inter-hand interval. If we look at the relationship of the P15, when we stimulate the left M1, we can see that there is a positive relationship. So that the shorter the conduction delay, the better behavioral performance. On the other side, also, the conduction delay from the right M1 has an impact on behavioral performance, but this is in the opposite direction. So that the lower the conduction delay, the worse the performance. And if we create an index with the rate between left and right, this rate is actually the best predictor of behavioral performance. So to some extent, these data are in line with current theories on transcallosal conduction delay because they do show that in some cases a shorter conduction delay is optimal and it affects, it improves behavior. But on the other side, we also found this quite surprising effect that a long conduction delay can be, a short conduction delay can be detrimental for performance. So our data basically sustain that in some cases, a temporal advantage of one hemisphere, of the information from one hemisphere to the other might be actually more beneficial than a general faster signal transfer between the hemispheres. To summarize, we have evidence that TMS-EEG can provide a measure of conduction delay of callosal fibers. We also found evidence that the transcallosal conduction delay can impact behavior in a task that requires high timing accuracy. Finally, we found that asymmetry of conduction delay was a predictor of better performance. Specifically, the asymmetry was that the signal from the dominant hemisphere had a temporal advantage over the signal from the non-dominant hemisphere. And this might, well, we can speculate that this temporal advantage is beneficial for the dominant hemisphere to have a better control during bimanual coordination. And that's the end. I would just want to thank all my collaborators who are working with me on these projects. And I want to thank you for your attention.
Hi, I'm Ida Grane from Aalto University, and I'm going to talk about how the pre-stimulus phase of spontaneous cortical oscillations affect signal propagation after a TMS pulse. So I will first go through some background for this phenomenon and then delve into the methods and results of this study. When you stimulate a cortical region with transcranial magnetic stimulation, the initially local responses first spread within 5 to 10 milliseconds to adjacent cortical areas and then to contralateral its areas after about 20 milliseconds. And this has at least been shown for stimulation of the primary motor cortex, shown in this upper row here, and the occipital cortex, shown in the bottom row. And this is shown with TMS combined with electroencephalography, so EEG. And the EEG signal can be taken to the source space to more accurately localize the activity. What this means is that the TMS-induced activity propagates to distant cortical areas from the stimulated region, and also this can be assessed with EEG. And individual responses to TMS are highly variable, both within the subject, but especially between subjects. And this is partly because the responses are modulated by the prevailing brain state during stimulation. So an example of this is the breakdown of cortical effective connectivity during sleep. TMS-evoked responses in awake subjects produce these high-frequency complex responses that spread to distant cortical regions, while TMS-evoked responses in subjects in non-REM sleep stay local and are of low complexity and quickly die down. And TMS-evoked responses are also modulated by spontaneous cortical oscillations. So both phase and power of the oscillations, as you can see with EEG, has been shown to modulate many aspects of the TMS-evoked responses. So as an example, the phase of the mu rhythm at the primary motor cortex predicts cortical spinal responses to TMS, or more specifically, stimulation during the negative peak of the oscillations results in bigger motor potentials and stimulation at the positive peaks of the oscillations. And in this study, we were interested in how the phase of the cortical oscillations affect TMS-induced signal propagation, meaning how stimulation during different local cortical excitability states might affect where the TMS-induced activity spreads after the TMS pulse. And the hypothesis is that the signal will spread to different distant cortical regions from the stimulated site, depending on the pre-stimulus phase of the mu and or the beta rhythms at the stimulation site. So to investigate this, TMS was applied to 2 cortical areas for 3 subjects, to the primary motor cortex, so M1 at the abductor pollicis brevis hotspot on the left hemisphere, and to right pre-supplementary motor areas, so pre-SMA. And the TMS-evoked responses were measured with a 60-channel NextStim EEG system. The stimulation parameters were fine-tuned to minimize muscle contractions, to keep the data as clean as possible, while keeping the early evoked responses at peak-to-peak amplitude of about 6 to 10 microvolts, and 250 pulses were given to each area. And after pre-processing of the data, the phase of the oscillations of the mu rhythm, so 7 to 13 hertz, and the beta rhythm, so 13 to 30 hertz, at the stimulated site were evaluated by using a narrowband Hilbert transform approach, where the pre-stimulus data segment is first padded with 500 points. Then you apply a Butterworth filter to this data and then a Hilbert transform to obtain the instantaneous phase exactly before the TMS pulse. And the trials were after this divided into 2 classes based on the phase, into a positive phase class and a negative phase class. And this figure here illustrates that if a TMS pulse had been given in the blue time window, this would have gone into the negative phase class, while a TMS pulse at the red time window would have made it go into the positive phase class. And this data here is otherwise. This resting state EEG data from one channel to illustrate this. And because the trials are sorted according to phase, some background oscillatory neural noise might still be present in the class average, which means that there could be a causal relationship between the responses and the phase due to the ongoing oscillations. To make sure any possible differences between the classes don't stem from this, pseudo trials were created from the EEG data between the TMS pulses. Then these trials were sorted according to phase, and their mean responses were subtracted from the actual trials. Then cluster-based statistics was used to assess the differences between the two classes. So for those not familiar with this method, it's used for solving the multiple comparisons problem, which is often a problem with EEG data, by clustering. So first you use a statistical test of choice on all data points. So you used an independent sample t-test as this initial test statistic. Then you cluster together the initially, so at this point, significant data points based on temporal and spatial adjacency, and add all the test statistics within each cluster to form a cluster level statistic. Then permutational tests are performed by reassigning the trials randomly into new classes a big number of times. And then the clusters are calculated for these new random classes. And you gather the cluster level values from these into a distribution. You can compare your original clusters with these to be able to assign p-values to the original clusters. And after setting a threshold for the p-values, you're left with only statistically significant clusters. So for the results, of the high variability between the subjects, as significant clusters were found at different time windows and scope areas for different subjects. And the mu rhythm seemed to play a big role at the stimulation of the primary motor cortex, as more differences in the TMS about EEG potentials we found for the mu rhythm than for the beta rhythm, while both rhythms seem to play an about equally big role of the stimulation of pre-SMA. So here's an example of the class differences for one subject after M1 stimulation for the mu rhythm. So the upper row shows when and where significant differences were found for the two classes, while the second and third rows show the average topographies of the positive phase class and the negative phase class. And the blue areas show that the negative phase trials have significantly larger EEG amplitudes than the positive phase trials. Yellow areas indicate that positive phase trials have significantly larger EEG amplitudes than the negative phase trials. And this you can also see from the topographies. So for example, let's get this blue area here. So the negative phase class have this strong red area that is either a lot weaker in the positive phase trials or possibly completely missing. And the topographies give a rough estimation of the locations of the active sources in the brain. And so these differences between the classes indicate that different brain areas are active at these times. So therefore, this indicates that the TMS-induced activity has spread from the stimulated area to different cortical areas, depending on the phase. So this seems to mean that the pre-stimulus phase modulates TMS-induced signal propagation, which means that the stimulated neuronal population prefers different connections based on the pre-stimulus local excitability. So there's a possible role of cortical oscillations in effective connectivity, which means that the cortical oscillations have extended roles past that of local excitability, and this also provides general further evidence on the influence of spontaneous cortical oscillations in shaping the neural responses to a TMS pulse. However, more investigation is still needed, so we need more data and more subjects to quantify the results and to understand how the pre-stimulus brain activity shapes the cortical responses to TMS. And we should also likely adopt some new methodology
So, for example, it's possible that the correction method for the background neural activity didn't perfectly remove this oscillatory noise before the statistical comparison of the negative and positive phase classes. Instead, the TMS rails could, for example, be compared with resting state data. So we're sure that we're looking at activity that's evoked by TMS, or use a more reliable method for removing the background oscillatory noise, such as a signal space projection source-informed reconstruction approach, so SSP-SIR for short. These results still give new insights into the role of cortical oscillations, and it's especially important for future closed-loop and brain state-dependent TMS paradigms, which reduce variability in studying cortical networks. And the possible future closed-loop stimulation paradigms will be vital for individualization of clinical applications of TMS, as well as for making treatments more efficient. And at the same time, this also opens up many new avenues of both clinical and basic research for further studying where exactly the signal propagates, what modulates this on a neural level, and more rhythms and brain areas could be studied as well. I want to thank especially my supervisors, Pantelis Tigamis and Tuomas Mutanen, and my other collaborators, Professor Mario Sanova, Aina Tervo, Victor Sousa, Jaakko Nieminen, and Professor Risto Ilmaniemi for all their contributions to this study. Thank you for your attention, and I will see you in the discussions.
Hello everybody, my name is Timo Roine and I will present today the Connect to Brain project. In this project we will develop a multi-locus transcranial magnetic stimulation device capable of connecting to the networks of the human brain. This project is a ERC synergy project of Aalto University Finland, University of Chieti Pescara Italy and University of Tübingen Germany. And its duration is from 2019 until 2025. Transcranial magnetic stimulation activates the brain non-invasively through electromagnetic induction. It is used for pre-surgical motor and language mapping and for treatment of neurological and psychiatric disorders, for instance depression. However, the current technology has several limitations. It can be only used to stimulate a single location, and changing the locus of the stimulation is very slow and operator dependent. It is also blind to ongoing brain activity. These limitations might lead to relatively low therapeutic efficacy of the current treatment protocols. To address these limitations, in this project we will improve the state-of-the-art brain stimulation protocols by developing multi-locus TMS devices able to electronically change the locus, direction, intensity and timing of the stimulation in real time. In addition, we propose to take into account ongoing brain activity measured by EEG to determine when and where to stimulate the brain next. This form of stimulation is called closed-loop stimulation, and we expect that it will result in much lower variability and a much larger effect size. To reach the goal of Connect to Brain project, we will need multi-locus TMS, real-time closed-loop system, and algorithms for the characterization of brain state, activity and connectivity. But most importantly, we need to translate these methods into clinical practice. In this video, we show an illustration of the proposed technology. The stimulus can be moved fast and reliably without mechanical coil movement. In addition, multiple spots can be activated, also simultaneously. EEG and other signals are measured to allow computer algorithm to design and perform a sequence of stimulations in closed loop. The project is divided into 3 parts, all of which work together towards the final goal, which is multi-locus TMS therapy. The 3 parts are all closely linked together. For instance, we need neurophysiological models to know what kind of brain state we need to reach to be able to develop this kind of algorithm and therapy. But we also need algorithms capable of estimating the brain state in real time to inform these neurophysiological models. In the closed loop, we start from high-density EEG data, from which we estimate brain source activities with inverse method. Then we estimate connectivity metrics, for instance by phase-based metrics, and we can use prior information from MEG and MRI for both of these steps. The time required for this part of the loop will be crucial. The parameters and brain states need to be estimated to be used in a machine learning framework which will define the next stimulation parameters for the multi-locus TMS device. In summary, the closed-loop system should be able to measure and predict the brain connectivity and state, and control the brain towards a desired state using real-time artificial intelligence methods. In the development of the multi-locus technology, our approach is to increase the number of TMS transducers by placing them over each other in several layers instead of side by side. This way, each coil produces a basis function for the electric field. By combining these basis functions or components, we can move the locus of the stimulation millimeter by millimeter and also change the direction of the induced electric field. The first prototype will consist of 5 coils and is capable of moving the locus of stimulation within an area of 3 by 3 centimeters and changing the direction of the field. However, the final prototype might contain up to 50 coils to be able to stimulate almost anywhere on the cortical surface. In addition, we have developed real-time tractography-based neuronavigation, which can estimate the structural connectivity of the stimulation site in real time. This can be used for the neuronavigation in this system. This method is presented in more detail in the poster presented by Dr. Eidogan tomorrow. The ability of the multi-locus TMS system to electronically control the stimulation location and orientation will also allow, for instance, automatic algorithms for the target search. These provide an operator-independent way to find, for example, the motor hotspot. On the right, there's an example of the stimulations performed by the algorithm in order to find the optimal site, marked by the X. The Tübingen group has demonstrated the feasibility of closed-loop TMS by analyzing EEG in real time and accurately predicting the occurrence of a specific brain state. In this case, the next peak or trough of the mu rhythm over the sensory motor cortex. These conditions represent states of low and high excitability measured by the motor evoked potential signs. Stimulating repetitively the same brain state results in long-term depression or potentiation-like plasticity. This finding is very important for our project. The same stimulation protocol leads to opposite change in the brain. So the outcome critically depends on what brain state the stimulation was performed in. And it is the brain state primarily that affects the outcome. In addition, the Tübingen group has shown that the phase synchronicity of the conditioning stimulus and test stimulus in the motor cortex plays a role regarding the intracortical inhibition. These 4 conditions occur very often in the brain, and thus the real-time system is able to wait for the optimal conditions to occur. And this specific experiments show that the inhibition is stronger for the in-sync condition. In summary, the Connect-to-Brain device will be a revolutionary instrument capable of stimulating the brain in completely new ways. It will be a game changer in treating brain disorders by improving the therapeutic efficacy. It will also provide novel insight into human brain functions. And if the translation to clinical use is successful, it will also massively reduce public health costs. Finally, this is a group photo of most of the personnel working in the project from Aalto University, University of Chieti-Pescara, and University of Tübingen. Thank you for listening.
Good morning to all who are following the BrainSTIM 2020 conference. My presentation today will focus on some practical aspects regarding the estimation of EEG connectivity in real time. In particular, these aspects are related on the amount of data which is required to obtain reliable connectivity estimates. The work that I'm going to show in this presentation was carried out in the framework of the Connect to Brain project. This is a ERC synergy-funded project whose aim is to develop new technology for multi-locus TMS combined to real-time EEG in a closed-loop system for neuromodulation therapy. So I will start from this point. Concurrent use of EEG and non-invasive brain stimulation techniques, in particular TMS, has received an increasing attention in the last years. One of the most interesting applications is combined EEG TMS in a closed-loop approach. A closed-loop approach means that the EEG recordings are processed online in real time to check the modulations of brain activity induced by stimulation. And this information is used to dynamically adjust the stimulation settings. This approach has recently led to the concept of a brain state-dependent stimulation, for which the TMS pulses have to be delivered at the right time at defined brain states, which are measured by real-time EEG in order to maximize the effect of stimulation. So far, the protocols used for closed-loop simulations mainly relies on the analysis of EEG activity, mainly based on information coming from amplitude or phase of the EEG signal. Recently, there has been an interest in exploiting information coming from EEG connectivity. EEG has a sufficient temporal resolution to directly investigate the functional coupling between brain sources, and has been successfully used to investigate brain connectivity and functional organization of the brain into networks at different time scales. In this respect, the use of real-time EEG connectivity to inform TMS in a closed-loop system represents a new avenue for closed-loop stimulation. So we can track changes of not only activity but also on the connectivity between sources in real time. However, the estimation of connectivity in real time is somehow challenging. In our group, we are investigating to what extent those techniques which have been used to estimate connectivity in offline modes can be extended to the real-time domain. Those techniques are mainly based on a sliding window which is moved in time, and connectivity is estimated within every window. And this results in time courses of connectivity. And the length of this window is somehow determines the resolution with which we can obtain these time courses. So in order to track fast changes of connectivity, a short window length is desired. And with this study, we essentially aim to quantify which is the minimum window length which can be used while still keeping reliable connectivity estimates. In order to answer this question, we use simulations. We simulated the activity for pairs of interacting sources. So we used a phase coupling model to simulate the activity of these sources. We also simulated the activity of noisy sources as background brain activity. From the source signals, we simulated the EEG recordings. We also added some sensor noise, and then we went back to the source space by reconstructing the activity of the sources. From the reconstructed source activity, we estimated the phase of the signals and then phase-based connectivity metrics between these signals. There are two main approaches to the analysis for the estimation of the phase. One is the analytic representation of the signals based on the Hilbert transform, and the other one is the Fourier representation. With the former, we calculated connectivity metrics such as the phase locking value, the imaginary part of the phase locking value, the phase lag index, or the weighted phase lag index. With the second one, we calculated these four metrics and additional three metrics based on coherency, which were originally defined in the Fourier domain, such as the magnitude of coherency, the imaginary part of coherency, and the lagged coherency. For the coherency and the phase locking value, we also orthogonalized signals before calculating connectivity, for removing the effects of source leakage. So this O stands for connectivity calculated on orthogonalized signals. Next, we tested the obtained connectivity metrics by comparing their values to an empirical null distribution obtained by using surrogate data. If the connectivity values obtained from signals exceeded a threshold value, it was considered significant. And we used this approach to evaluate the performance of the different connectivity metrics for various lengths of the signals, which are comparable to the length of the windows which are commonly used in dynamic connectivity analysis, so for short windows in the order of hundreds of milliseconds. We also performed thousands of simulation repetitions at different levels of noise. Now, I will first discuss the results that we have obtained for connectivity metrics based on the Hilbert transform. This plot shows the box plots of the distributions of the connectivity metrics as a function of the data length L, measured in number of cycles of the oscillations at the frequency of interest. For instance, in these simulations, we simulated a 10 Hz activity, so 1 cycle is equal to 100 milliseconds. You can see that for decreasing data length, there is an increasing bias, which is denoted by the fact that the data from the null distribution in red, data without interaction, become quite large. This means that if you use short windows, even signals which were not interacting seems as to be interacting. And therefore, at a short window length, it is difficult to recognize a true positive from the TESSA distribution from a false positive from the null distribution. For evaluating the performance of the metrics, we use the ROC curves. In particular, this plot shows the area under the ROC curves for the 4 connectivity metrics as a function of the data length. You can see that for large values of the data length, the connectivity metrics show all large values of the area under the curve that is quite close to 1, which means a not optimum, an excellent performance of these metrics. However, for decreasing data length, this value decreases toward small values. The values below 0.7 are often regarded to as poor performance of the method. And if we choose a reference value, for instance, 0.8, which is often regarded to as a value which denotes a good performance for the methods, it represents the probability that a random value chosen from the random true positive is ranked larger than a true negative. So if we choose this value, we found that if we need at least a value between 4 and 7 cycles at 10 decibels, which for a 10 Hz oscillations means a value between 400 milliseconds and 700 milliseconds, while a number of cycles between 5 and 8 at 3 decibel, and or a value between 6 and 8 cycles at 0 decibel. Also, the value of the area under the curve represents the performance of the metrics and therefore can be used to compare the performance of the different connectivity metrics. We found that while at large data length, the area was, for all the connectivity metrics, was quite similar, which denotes a similar performance, these values become quite different at short data length. This means that at short data length, the choice of the connectivity metrics matters. For instance, we found in these simulations that PLV performed better than all the other connectivity metrics. Now I will show the results obtained for connectivity metrics based on the Fourier transform. And for these, the area under the curve is a function of both the data length L as for the previous case, but also as a function of the window length which is used to calculate Fourier coefficients. Because for calculating cross spectra, for instance, the Fourier coefficients have to be calculated within the signals, have to be chunked into segments, and the Fourier coefficients have to be calculated within every window, and cross spectra are calculated by averaging the information from different windows. We found a common pattern for all the connectivity metrics that we discussed for the PLV. We found that the area under the curve increases for increasing data length and for decreasing window length, the window which is used for calculating Fourier coefficients, apart for very small values of the window, which I will comment in 1 minute. This is fine, but this poses two limitations, because in real time, in dynamic connectivity analysis, we are interested at short data length, and also if we decrease the window length, we lose the frequency resolution. So if I'm interested to perform the analysis with a 1 Hz resolution, for instance, I cannot go below 10 cycles at 10 Hz, for instance. 10 cycles means a 1 second length, which for W, which determines a frequency resolution of 1 Hz. So this is also the reason why at very small values for the window length we have a decrease of the performance of the methods, because the frequency resolution is so poor that the signal of interest is contaminated by noise, by the activity which is outside the band of interest. At least this value, the corner to which the area that the curve started to decrease, depends on the frequency content of the signal. In our simulation, we simulated a 10 Hz activity for a 2 Hz bandwidth. So if we go below 5 cycles, so if the frequency resolution is larger than 2 Hz, so we have a contamination of noise, and so the performance of the matrix decreases. These are the counterplot for the area under the curve obtained for all the connectivity metrics. And so also in this case, if I choose a reference value of 0.8, and if, for example, I want to perform the analysis with a 2 Hz resolution, so I have to use a 5 cycles window length. To achieve a 0.8 area under the curve, I need a value between 7 and 11 cycles of data length at 10 decibel, or between 8 and 12 cycles at 3 decibel, or a value between 9 and 14 cycles at 0 decibel. So values which are larger compared to the values which were observed with Hilbert-based connectivity metrics. So to summarize the results that I've shown, in order to answer my question of which is the minimum window length, we found that at least a few cycles of the oscillation at the frequency of interest for the window length are required to obtain reliable estimates. For instance, we found that for the PLV, to obtain a 0.8 area under the curve, we need at least 4 cycles, with 10 Hz means 400 milliseconds. And this value represents the minimum, determines the intrinsic temporal resolution with which we have the estimates of time-varying connectivity. Also, we found that Hilbert-based metrics showed a better performance than Fourier-based metrics for short window. The former allowed to, at least compared in terms of the area under the curve, has a similar performance than the latters for window lengths which are 1/3 or from 1/2 to 1/3 of those observed with Fourier-based methods. Also, and finally, the connectivity metrics, an approach to dynamic connectivity analysis based with a sliding window, can also improve by adaptive algorithms, which can be efficiently implemented also for real-time analysis. So in conclusion, our simulations show that we can somehow push the resolution of connectivity metrics toward a small value which allow a reliable use of EEG connectivity in real-time analysis. And this can be used for a more complete characterization of the brain state, so based on not only on brain activity, but also on brain connectivity, which can be used in closed-loop system for a closed-loop brain state triggered stimulation. I would like to thank people for my group, research group in Chieti, in particular Laura Marzetti, Vittorio Pizzella, Alessio Bassi, and Roberto Guidotti, which contributed to this work, and also the PI of the Connect to Brain project, Risto Moniemi, Gianluca Romani, and Ulf Zeman, and also all the members of the Connect to Brain project. And also, I would like to thank you all for the attention.
Hello, I'm Mikko Losterinen. I'm a doctor here at the Helsinki University Hospital at the Department of Clinical Neurophysiology. And the study I'm introducing for you today is a part of a project of collecting reference values for motor evoked potential exams at our department. The motor evoked potential exams, or MEP exam for short, use transcranial magnetic stimulation, or TMS, to elicit peripheral motor responses and can be utilized in diagnostics to assess the function of a patient's motor pathway, for example, when upper motor neuron disease is clinically suspected. Diagnostically, the single most useful variable obtained in an MEP exam is the central motor conduction time that has been shown to increase in central motor pathway lesions. It can be measured with single pulse TMS by first measuring the total latency between cortical stimulation and distal motor response, and then subtracting from this the time between nerve root level stimulation and distal motor response. By contrast, the amplitudes of MEP responses are not as useful in diagnostics, because they have a lot of inherent variation between individuals and also between tests. Indeed, many factors can either facilitate or inhibit the MEP responses that depend on the pre-stimulus excitability levels of neurons in the motor cortex. Using a paired pulse TMS, it's also possible to assess this so-called intracortical inhibition and facilitation during the MEP exam. In paired pulse TMS, a sub-threshold conditioning stimulus is first given below the patient's motor threshold prior to a larger supra-threshold test stimulus that is then intended to elicit an MEP response down the motor pathway. The amount of intracortical inhibition or facilitation you'd observe with this depends on the interstimulus interval between the two stimuli. Inhibition has been shown to be most prominent after 2 to 3 milliseconds of the conditioning stimulus, and facilitation peaking at about 12 to 13 milliseconds after the conditioning stimulus. The facilitating paired pulse can also be used in cases where you are unable to elicit reliable MEP responses from single pulse TMS, even with the maximum intensity of the magnetic stimulator. However, in clinical settings, our experience has shown that sometimes even with paired pulse stimulation, it's sometimes hard to elicit MEP responses from some patients. This is particularly the case in lower extremities, in older patients, and in patients who have a pathology of the motor pathway. So, another way to facilitate the MEP responses is pre-activation. In other words, the patient voluntarily contracting the target muscle while given the TMS stimulus. The amount of facilitation depends on the intensity of the muscle contraction and can be even more effective at facilitating the MEP response than paired pulse. But in clinical setting, there are sometimes problems when using pre-activation technique. Firstly, it requires the patient to be able to control the target muscle with somewhat precision, which is not always the case. Secondly, because of already present muscle activity at baseline, it can be difficult to accurately evaluate the exact starting point of the MEP responses, therefore possibly resulting in less reliable estimates of the MEP latency and of the central conduction time. So, in this study, we aimed to solve this problem by using 2 sub-threshold conditioning pulses paired prior to a third supra-threshold test stimulus. We call this technique the double facilitating triplet TMS. The idea is that by giving 2 conditioning stimuli with a suitable interstimulus interval below the motor threshold, you can further increase the amount of facilitation compared to a simple paired pulse with only 1 conditioning stimulus. Using a technique like this has been prescribed earlier, at least in 1996, by Zeman, Rothwell and Redding, but to our knowledge, its use or benefits in clinical diagnostics have not been reported before. This also should not be confused with the triple stimulation technique, which uses two electric peripheral nerve stimulators and a collision technique, together with TMS. Going over the technical details of our study, we had a sample of 50 volunteers who were between 24 and 70 years of age and healthy in the sense that they had no epilepsy, no medications affecting the central nervous system, and no pathologies of the central or peripheral motor pathway that we knew of. The test equipment used was a Maxim 3-into-1 stimulator with a monophasic pulse, a round coil, and maximal power of 2 Tesla. MEPs were measured from target muscles with surface electrodes using a clinical EMG system. The abductor pollicis brevis was used as target muscle in the hand and the tibialis anterior in the leg. Electric stimulations to median and peroneal nerves were first used to exclude peripheral nerve block. Then, with single pulse TMS, the optimal transcranial stimulation spot was located and the motor threshold value, later called MT, was determined as the lowest stimulation intensity that produced MAP responses with peak-to-peak amplitudes of over 50 microvolts in at least 5 out of 10 stimulations. 5 single pulse measurements were then carried out with stimulation intensity relative to 120% of MT. In addition to single pulse measurements, 5 measurements of facilitating paired pulse and facilitating triplet pulse were conducted. An interstimulus interval of 13 milliseconds was used between all pulses, with stimulus intensities of 90% MT for the conditioning pulses and 120 of MT for the test pulses. Mean amplitudes were then calculated from each 5 measurements, including possible zero amplitude ones. Okay, so the results of the study showed that triplet pulse TMS produced MEP responses of significantly higher mean and median amplitude than either single pulse or paired pulse TMS in both target muscles. The increase in amplitude was more substantive in the leg. Also, we observed 2 instances in our healthy test subjects that a single or paired pulse TMS failed to produce any reliable MEP responses, but triple pulse succeeded. In this graph, you can see the mean amplitude distribution of each 5 measurements with single, paired and triple pulse in both target muscles. Outlier values are marked with dots, the middle line is the median, and boxes represent the upper and lower quadrants of the distribution. All of the differences between the mean amplitudes were statistically significant. Additionally, we observed that the amount of facilitation on MEP amplitudes between paired pulse and triple pulse had a strong, almost direct correlation. This was the case in both the upper and lower extremities. In these graphs, you see function of the relative amplitudes compared to single pulse amplitudes of paired pulse and triple pulse. And as you can see, on an individual level, the relative rise in amplitude between paired and triple pulses is quite comparable. Also worthy of note is that both paired pulse and triple pulse TMS significantly shortened the MEP latency compared to the baseline single pulse stimulation at 120% of MT. Here you can see the mean latency times in single, paired and triple pulse measurements for both target muscles. So, from the results we conclude that in this relatively large sample, facilitating paired pulse TMS and facilitating triplet pulse TMS showed to be more efficient in producing MEP responses than single pulse TMS without pre-activation. Furthermore, the double facilitating triplet pulse TMS can be applied in the clinic and might even be a more reliable method than paired pulse. The amount of facilitation observed seemed to be correlating with paired pulse individually, so the double facilitating triplet pulse TMS could probably be used to assess intracortical facilitation as well. It also could possibly offer some novel diagnostic correlations, but further clinical testing on patients would be needed to study this. It would also be interesting to do a study comparing the latency estimations and amplitude values of MEP responses when using the triplet pulse TMS or the pre-activation technique to produce facilitation. Finally, when comparing results to reference values obtained with single pulse stimulation, the shortening effect on MEP latency should be accounted for when using double facilitating triplet pulse MEPs.
Okay, thank you for your time. I hope you were at least somewhat brain stimulated.
My name is Nikiforou Elizaveta. I'm from Center for Cognition and Decision Making at the Higher School of Economics, Moscow. And I'm a member of the neurodynamics team where I work with Pavel Novikov, Maria Nazarova under the supervision of Vadim Nikulin. And I really hope that all of you doing well during these challenging times. And anyway, today I would like to present to you our latest project. Supplementary motor area is a target zone for non-invasive neuromodulation. A systematic review of basic and clinical approaches. So today there are still different neuropsychiatric syndromes that remain pharmacoresistant, and one of them is catatonia, for which the electroconvulsive therapy is still the most effective treatment, even now in the 21st century. And it seems to be obvious that new approaches, less invasive, are necessary to be developed and invented to treat those patients. And repetitive transcranial magnetic stimulation and transcranial direct current stimulation could be those approaches. But there are still no protocols and no established protocols to treat those patients, and there are only several cases in the world today. So, and the important thing that supplementary motor area was discussed as a promising target for non-invasive neuromodulation in catatonic patients. Why supplementary motor area? What is supplementary motor area? So, supplementary motor area is a multifunctional brain area that is believed to be involved in many motor and cognitive functions. Supplementary motor area was described 70 years ago as one big area lies anterior to the primary motor cortex, but today it is not divided brain areas in other sources into 3 brain areas, distinct brain areas as supplementary motor area proper. You can see it here. Pre-supplementary motor area and supplementary eye field. Area proper, pre-SMA, are functionally different, but it is that they lie next to one another, and it is hard even anatomically to use them, and contemporary that dot anterior commissure, and that's where the border between those brain areas lies today. And so it gets challenging when you try to stimulate pre-SMA, not to disturb the brain area line next to it. And also it is important that supplementary motor area has a symmetric representation of the body, and it is with basal ganglia also symmetropic manner. The association is associated with basal ganglia and symmetropic manner. And repetitive transcranial magnetic stimulation of the supplementary motor area seems to be promising in 3 diseases. It is Parkinson's disease, obsessive compulsive disorder, and Tourette syndrome. So relying on all this information we get, We aimed with this system review, we aim to provide a comprehensive overview of the SMA functional properties that were examined with RTMS. And we suggest that such systematic review would be helpful to develop new non-invasive protocols for neurological disorders as catatonia that still remain pharmacoresistant, and there are no not really invasive treatments. So the methods we used in our systematic review, we followed and Prisma guidelines. We registered our systematic review in the Prospera system. Here you can see our registration number. Cochrane and Scopus that were available up to 27th of 2019. Also, 3 investigators assessed studies quality with Cochrane risk of violence tool, and all articles were divided into 2 categories. It was the study with healthy volunteers and studies including patients. And qualitative analysis was performed using narrative textual synthesis. Here you can see the study filtering process, so the flowchart. We downloaded 808 articles from the 3 mentioned databases. And after all filtering, we get left with the 92 articles we included into our analysis. Half of them were studies only with healthy participants, and another half included patients. So let's proceed to the results of our qualitative analysis. And first of all, let me show you the studies with only healthy participants that had positive outcomes with SMA as a target for RTMS. First, you can see that functions were examined as using inhibitory RTMS protocols, excitatory RTMS protocols, and articles were divided into 2 big groups: motor control and cognitive functions. And here you can see that the most functions that were successfully modulated, as with inhibitories or with excitatory protocols, were successfully modulated sequence processing, and motor inhibition, movement preparation, and bimanual coordination. Those we highlight as the most frequently studied functions SMA with RTMS. And I would like to highlight the emotional process, the process of emotions. Speaking of the pre-SMA studies with only healthy participants, well, we can see that it is much less studied than SMA, but also excitatory protocols were used, and were examined 2 motor control functions and 1 cognitive function of the SMA, and the most studied functions, 7 studies successfully motor vision with the RTMS SMA stimulation. And now let's continue with the studies with positive outcomes, where SMA was modulated by RTMS in patient groups. First, yes, also as inhibitory protocols were used. SMA was chosen to target in Parkinson's disease, obsessive compulsive disorder, Tourette syndrome, autism spectrum disorder, bipolar affective disorder, and chronic pain syndromes. We can see that Parkinson's disease is the most investigated, the most examined disease with RTMS, SMA RTMS. And I would like to highlight that inhibitory protocols were used to reduce those symptoms and those of symptoms that are known as hyperkinetic. And I would like to underline once again that it is only positive outcomes. So in those studies, investigators succeed use those symptoms. And excitatory RTMS was used to reduce hypokinetic symptoms and actually succeed to actually improved performance patients with self-initiated movements and with handwriting, improved handwriting of Parkinson's disease patients. There were 6 studies, 6 articles of where investigators succeeded to reduce compulsive symptoms, obsessive compulsive disorder, and also were investigated succeed to reduce. And speaking of other of other studies, of autism spectrum disorder, bipolar affective disorder, and chronic pain syndrome. As for pre-SMA, there were only 2 positive studies where investigators succeed to reduce obsessive compulsive disorder symptoms, and also with inhibitory RTMS, investigators and excitatory TMS reduced restless leg syndrome. On previous slides, I showed you only articles with positive outcomes, and here I would like to show you the overview of number of studies and how many of them were positive, how many of them were negative. And here you can see studies with healthy participants, and here you can see the studies with analysis not only studies with that got significant outcomes, but also the negative studies. And at the end, I would like to say that our analysis highlighted and revealed very important functions of RTMS that would help further investigators and further medical doctors to invent new protocols, and as SMA functions as sequence processing, motor repair. Most importantly, that SMA was shown to be involved more studies for modern inhibition than SMA. And speaking of clinical application, SMA actually seems to be a promising target for neuromodulation in patients with affected system, especially when we're talking about Parkinson's disease, obsessive compulsive disorder, and Tourette. Of course, we combined and systematized what was already known, but this systematic review, we hope when our investigators and doctors develop new neuromodulatory RTMS protocols for such resistant as catatonia. Thank you for your attention.
Hi everyone, I hope you're all doing well and stay safe at home. My name is Manon Desforges. I'm doing my master degree in psychology at University of Montreal, and today I'll be talking about TMS-EEG as a measure of the intermittent theta burst stimulation mechanism in the prefrontal cortex. So in our lab, we're working on optimizing the treatment of major depressive disorder because more than 300 million people are living with depression in 2017, according to the World Health Organization. So it's about 4.4% of the world's population, and unfortunately, 1/3 of patients fail to respond to antidepressive therapy. So there is a real need for alternative treatments and measures to assess the effectiveness of those treatments. And this is exactly what we're working on. We are more specifically interested in the treatment of depression using a technique called TMS. So what is TMS? Transcranial magnetic stimulation is a neurostimulation tool where a coil placed over the skull stimulates the brain region underneath with a magnetic field. How does it work exactly? Well, according to Faraday's law of electromagnetic induction, a magnetic field generates an electric current. So, by applying a magnetic field over the skull through a TMS coil, we are also inducing an electric current, and this current will lead to a neuron's depolarization. And the magic of this tool is that we can use it in two different ways. Firstly, we can use it to measure the brain activity. For example, if we stimulate the motor cortex, then we can record the muscular activity with electrode placed on the person's finger. And secondly, when the TMS is applied repeatedly, it's then called RTMS or repetitive TMS. It allows to modulate brain activity beyond the stimulation period. And this RTMS technique is actually used as a treatment of depression. So let's talk about this RTMS treatment. Well, the RTMS standard protocol involves stimulation of the dorsolateral prefrontal cortex, or DLPFC, for 20 to 45 minutes from Monday to Friday for 4 to 6 weeks. And it's been shown to be effective to treat depression, but it's very time consuming. Fortunately, in 2005, a new form of RTMS was developed, the theta burst stimulation, or TBS. And this technique allows to reduce the stimulation duration down to 3 minutes and to stimulate at lower intensity. And the recent large trial showed comparable efficacy compared to the standard RTMS. So it's really promising. One of the different TBS protocol is the intermittent TBS, or ITBS. It consists of 3 bursts of 50 Hz pulses repeated at a 5 Hz frequency. And although it is already used in clinical practice, the optimal duration of stimulation is unknown. The standard ITBS protocol is 600 pulses, so it's around 3 minutes stimulation, but there is an increase in duration based on the assumption that it will increase the therapeutic effects. For example, some studies are applying 1200 pulses or even 1800 pulses. So the aim of this study is to determine the optimal ITBS duration to activate the left DLPFC out of the 3 most commonly used duration, 600, 1200, and 1800 pulses. So let's now talk about the methodology. We have recruited 14 right-handed healthy participants, half men and half women. Cohort average was around 26 years old. And I want to bring your attention to the fact that our participants were healthy controls. They were not diagnosed with depression, and it may look counterintuitive to test non-depressive people to assess the efficacy of depressive treatment, but the idea is that we firstly want to understand how the healthy subject brain works before applying it to depressive subjects. Here's the list of the equipment we used. A neuronavigation system was used to target the left DLPFC. The specific MNI coordinates were based on a previous study and corresponds to the area around F3 here. And the participant's brain was modelized thanks to a standard MNI brain model. Then we used a combination of TMS and electroencephalography, or TMS-EEG, to record the cortical activity. So TMS-EEG allows to quantify cortical excitability changes induced by TBS outside the motor cortex. So we are sending a TMS pulse and we record the electrophysiological response produced by the pulse with a 64 TMS compatible channel EEG cap, like this one here. The response evoked by the TMS, called TMS evoked potential, or TEP, consists of a series of positives and negative deflections lasting up to 300 milliseconds. TEP is composed of various characteristic cortical responses associated to components such as P30, N45, P60, N100, and so on. TEP can also capture additional neurophysiological information, such as the event-related spectral perturbation, or ERSP, when we can observe the change following ITBS in the time-frequency domain. Each participant underwent the 3 different durations of ITBS, so either 600 pulses, 1200 pulses, or 1800 pulses, during 3 different sessions, separated by at least 7 days. The session order was randomized and counterbalanced across participants. And for each session, we took TMS-EEG measures before ITBS. It was 80 single pulses. Then ITBS was applied, and then we took the same TMS-EEG measures after ITBS. About the TMS-EEG pre-processing, data were pre-processed with MATLAB, EEG Lab, and Filtrip. Data were epoch around the TMS pulse and baseline corrected. After noisy channel and epoch removal, we apply an interpolation for missing data. We also performed 2 rounds of independent component analysis, or ICA. The first ICA to remove the TMS artifact, and the second ICA to remove other artifacts such as eyes movement, muscle activation, and so on. And then we also apply a bandpass filter and a notch filter, and we finally re-reference data to the average of the scalp electrodes, of all scalp electrodes. All statistical analyses were conducted on a specific region of interest, including 5 electrodes, AF3, F1, F3, FT1, and FT3. We conduct 2 linear mixed models with condition and time as within subject factors, one for each TP of interest, so P30, N45, P60, N100, and P200, and one for each frequency of interest, so theta, alpha, beta, and gamma. A p-value under 0.05 was considered as significant, and 2 key post-hoc t-tests with bond-Ferroni correction were also conducted. So let's now talk about our results. So for TP, we found that none of the 3 stimulation durations was superior in modulating the amplitude of TPs. However, the 3 ITBS durations significantly modified the majority of the components. So x-axis represent the time in milliseconds, y-axis represent the amplitude in microvolts, y-axis is set at time 0 and corresponds to the TMS pulse. The green curve shows the activity of all conditions before TBS, and the blue one after TBS, and the star indicate a significant effect of time. So P30 is an index of cortical excitability, and our results suggest that ITBS significantly reduced its amplitude. N45 component seems to be associated with cortical inhibition, and our results demonstrate a significant increase in the amplitude of N45. It's a negative component, so it's reversed, suggesting that ITBS would increase the inhibition of healthy subjects. As P30, P60 seems to be associated with excitability, and our results show a decrease of P60 amplitudes following ITBS. And this would have promising clinical perspective, as a recent TMS-EEG study reported that P60 amplitude over the left DLPFC is significantly larger in depressive people than in healthy controls. N100 seems to be associated with inhibitory mechanism, and more specifically with GABA-B. Our results do not show significant differences in its amplitude, suggesting that the ITBS would not modulate N100 inhibition. Studies suggest that the N100 can be confounded with the auditory artifact, which may explain the lack of significant result, even though we used a white noise to mask the sound. And finally, P200 seems to be associated with cortical excitability. consistently with P30 and P60, a decrease of its amplitude is observed. And it suggests that the mechanism of prefrontal ITBS may differ from the motor cortex, where an increase of cortical excitability is observed. So now the ERSP results. So x-axis represent the time in milliseconds, y-axis represent the frequency in hertz, time zero represent the TMS pulse. On the left, you can see the activity of all conditions before TBS, and on the right, you can see it after. And black boxes represent our frequency band of interest with their specific time windows. So our results show that none of the three stimulation duration was superior in modulating the power of ERSP. However, the three ITBS durations significantly modified theta bands, where we observe a decrease of theta power following ITBS. For alpha, we found a trend for an effect of time suggesting that the power of the ERSP was reduced post-ITBS, and no significant results were found for beta and gamma. So a couple of things to remember about this talk. TMS can be both used as a treatment tool and as a measuring tool. TMS-EEG is a promising technique for determining the optimal parameters of stimulation of the DLPFC for clinical applications. Results show no difference between 600, 1200, or 1800 pulses, ITBS pulses, on the modulation of cortical excitability. It suggests that increasing the duration of stimulation may not be related to increased clinical efficacy, but this will need to be replicated in clinical sample. And yeah, so that was my master degree thesis, where we looked at the optimal duration of ITBS stimulation. And for my PhD thesis next year, I'm gonna look at the optimal paradigm of stimulation, so either unilateral or bilateral stimulation. And finally, I would like to thank Dr. Sarah Tremblay for amazing supervision, Dr. Itai Das for his precious help, Dr. Martin Lepage and Dr. Jeff Descalakis, as well as all the contributors of this project. And thank you for watching this video. Bye-bye, take care.
Good afternoon. My name is Silvia Casarotto and I work at the University of Milan. My talk is entitled TMS-EEG is sensitive to the brain's capacity for consciousness, a reproducibility study. Our brain is the engine of our actions, perceptions, feelings, and thoughts. To the extent that we produce a motor output upon a sensory input, we can say we are conscious. Conversely, when we lay in deep sleep or during anesthesia, we aren't conscious because we are unresponsive and disconnected from the external world, and upon awakening, we do not provide any conscious report. However, in some conditions, we can be conscious and unresponsive at the same time. This occurs, for example, during dreaming and ketamine anesthesia. In these conditions, in fact, we appear as unresponsive to an external observer, but upon awakening, we can provide vivid description of a weird conscious experience entirely produced within our brain. Now imagine to have a severely brain-injured patient. Because of lesions, this patient could be impaired on sensory input or motor output, or both sensory and motor pathways. How can we say whether the patient is conscious or not? This time we have to open the black box. But what to measure? There are several theories about this. But the rather common feature across theories that has been identified as important for consciousness is complexity of brain activity. That is, the brain's capacity to process information in an integrated manner. So let's start from this theoretical prediction. To the extent that functionally specialized brain regions work in synergy to integrate information across highly specific functional networks, thalamocortical networks, we are conscious. How can we evaluate whether the brain is actually working like this in a conscious individual? By combining transcranial magnetic stimulation and electroencephalography. That is, by recording the overall electrical brain activity evoked by a focal, non-invasive, and direct perturbation of cortical neurons with TMS. If we imagine to stimulate a node of such a network, we will obtain a local activation that travels towards distant sites and produces secondary activations with highly specific temporal dynamics. In this case, we are conscious. If because of physiological, pharmacological, or pathological reasons there is an imbalance towards loss of integration, the network becomes modular. So stimulation of a node of such a network will produce a local, simple and slow activation that does not travel elsewhere. In this case, we say does not allow to sustain consciousness. On the other side of the spectrum, if the imbalance is towards loss of differentiation, stimulation of a node of such a network will produce a global and stereotypical activation that spreads like wildfire. Also in this case, we say that consciousness cannot be present. These theoretical predictions turned out to be true when looking at EEG responses to TMS recorded in healthy awake subjects during disconnected consciousness, such as dreaming and ketamine anesthesia, and during unconsciousness, such as during deep non-REM sleep and propofol midazolam xenon anesthesia. But in order to make all this clinically useful, we have developed this Perturbational Complexity Index, PCI, PCI is a real number between 0 and 1 that can be derived from EEG responses to TMS. And this number measures the complexity of the spatiotemporal pattern of cortical activations significantly evoked by TMS. From the distribution of PCI values measured in a large benchmark population, we derived an empirical cutoff, PCI star, that discriminates with 100% sensitivity and specificity between unconsciousness and consciousness, even if disconnected. Now, what happens if we apply this empirical cutoff PCI star to the distribution of PCI values measured in severely brain-injured patients with disorders of consciousness? Let's start from minimally conscious state patients. These patients show minimal signs of purposeful behavior fluctuating over time. As you can see here, almost 95% of these patients is correctly detected by PCI, showing that PCI is extremely sensitive to minimal signs of consciousness, which are visible from behavioral evaluation. What about vegetative state patients? These patients are able to perform reflexive movements only. Well, according to PCI, this rather homogeneous clinical category can be further stratified into 3 subgroups. We have no response patients with PCI equals 0 who show no significant EEG responses to TMS. At the population level, most of these patients have anoxic etiology and show voltage-suppressed EEG background. Then we have low-complexity patients who show slow and stereotypical EEG responses to TMS. PCI value of these patients is below the empirical cutoff. Finally, and most importantly, we have high-complexity vegetative state patients who show complex EEG responses to TMS with PCI values well above PCI star. Now, this neurophysiological stratification of vegetative state patients has clearly ethical and therapeutic implications. And also, concerning the outcome, you see that no response patients have a worse outcome as compared to high complexity patients. So all these results are very nice and promising, and we would like to promote the application of PCI in the clinics for diagnosis as a prognostic marker and to longitudinally monitor recovery of consciousness in patients. But to do this, we need first to further confirm and replicate these nice results in larger patient populations with different etiology, different brain lesions, and across clinics. This is what we are doing within this project funded by the Italian Ministry of Health. This project involves 2 clinical institutions in Sicily with patients with disorders of consciousness. This is the TMS-EEG equipment we are using. I want to underline that this equipment is different from the one used in the previous validation study. So far within the project, we have collected TMS-EEG data in 5 patients only. Well, the project just started, and because of pandemia, we had to temporarily suspend the recordings. But we hope we can restart soon. All these patients were vegetative state patients, meaning that they could perform reflexive movements only. All of them except patient number 5. In this patient, it was possible to observe sometimes the capability of fixating an object. Fixation per se is considered a minimal sign of consciousness, but in this patient, it was very difficult to assess this capability. and it was not present all the times. And therefore, I must say that diagnosis for this patient is really borderline between vegetative state and minimally conscious state. What does PCI tells us about the consciousness state of these patients? Patients 1, 2, and 3 show PCI equals 0, with a flat EEG response to TMS. Patient number 4 shows a slow and stereotypical EEG response to TMS with PCI value below PCI star. Patient number 5 shows complex EEG responses to TMS with PCI value well above the empirical cutoff PCI star. Now we can conclude that in this rather small sample of patients, we were able to replicate the neurophysiological stratification of vegetative state patients described in the previous validation study. This successful replication has been obtained using a different TMS-EEG equipment and including patients with metabolic etiology who weren't present in the previous validation study. Last, as in the previous study, no response patients have a prevalent anoxic etiology and typically show a voltage-suppressed EEG background, whereas low and high complexity patients show a more preserved EEG background with faster oscillations and larger amplitude. Thank you very much for your attention.
Good afternoon. My name is Katri Silvennoinen. I'm a clinician training in neurology and currently working towards a PhD at UCL. Thank you very much for the opportunity to discuss our ongoing work on TMS evoked potentials in Dravet syndrome. I'll start off by providing some background on Dravet syndrome, as well as our rationale for using TMS-EEG to study this condition. I'll then outline our methodology and present preliminary findings for the small group of people that we've studied so far, contrasting these with healthy control data. Loss of function variants in the gene SCN1A are associated with a wide range of epilepsy phenotypes. This gene encodes the alpha subunit of the type 1 voltage-gated sodium channel. Generally, voltage-gated sodium channels are essential for the initiation and propagation of action potentials. This particular sodium channel is predominantly expressed in inhibitory interneurons, and indeed, animal models have shown that loss of function variants lead to impaired phasic GABA release, therefore supporting the idea that as a disease mechanism, a net decrease in inhibition is relevant. On the spectrum of SCN1A-related epilepsies, Dravet syndrome is at the severe end, and this is associated with typically de novo loss of function variants. It presents in a previously normally developing child with typically febrile seizures before the age of 12 months. Following this, seizures begin to occur also outside of the setting of fever and include numerous types, including generalized tonic-clonic, tonic, and myoclonic. Developmental delay becomes increasingly evident, and children have particularly problems with language and many have autism symptoms. In childhood, EEG may show generalized discharges. In adulthood, however, epileptiform activity is typically described as a focal or multifocal with a slow background, as shown here for one of our patients. There are no gross neuroimaging abnormalities associated with this condition. Epilepsy is often refractory, and unfortunately, patients are at high risk of epilepsy-related early mortality. Therefore, there's a pressing need for new syndrome-specific treatments and biomarkers for assessing the response and particularly predicting the response for such treatments. A previous study from our center using TMS-EMG showed a lack of normal short interval intracortical inhibition in a group of people with Dravet syndrome. CICI is considered to be a measure of GABA-A-ergic cortical inhibition, and therefore this finding is very much in keeping with what's understood about the disease mechanism. We've subsequently expanded this work and studied further individuals both with Dravet syndrome as well as some with milder SCN1A-related epilepsies. The findings suggest that among those with pathogenic or presumed pathogenic SCN1A variants, the GABA-A-ergic impairment, as measured by TMS-EMG CICI, is specific to those with severe phenotypes. So on this background, we hypothesized that TMS-EEG might shed further light on the neurophysiological abnormalities underlying this condition. Given that TEP components have been shown to be under GABA-A-ergic modulation, as a gene-specific hypothesis, we expected that loss of function in SCN1A could lead to abnormal TEP morphology. Secondly, as an epileptic encephalopathy associated with neuropsychiatric symptoms across multiple domains, we hypothesized that perturbation by TMS might reveal widespread abnormalities in brain activity, perhaps also overlapping with findings in other neuropsychiatric conditions. In terms of our lab setup, we use a monophasic MagStim stimulator with a bigger 8 coil. We have a 64-channel cap with active electrodes and a BrainVision ActiChamp amplifier. For neuronavigation, we use a BrainSight system. For noise masking, we play a white noise through earphones, which is composed from stimulator clicks with randomly interspersed realistic click sounds. Stimulation was delivered in the left premotor area. The intensity ranged from 75 to 100% maximum stimulator output, but both the coil position and stimulation intensity were adjusted individually to reduce the artifact and to maximize the amplitude of the response based on visual inspection of evoked potentials calculated in real time. Unfortunately, we had to use templates in neuronavigation as the patient didn't have available up-to-date MRI suitable for this purpose. For one patient, a noise masking was abandoned due to discomfort. An ICA-based pre-processing was done using the EEG Lab Toolbox and scripts. So far, we have data for 3 patients. All have confirmed pathogenic SCN1A variants in keeping with their clinical diagnoses. They all have highly refractory epilepsy and significant learning disability and language impairment, and they take 2 to 3 antiepileptic medications. None have any known relevant neuroimaging abnormalities. I'll first show a demonstrative response to premotor stimulation from one of our healthy controls. The butterfly plot at the top shows the average response for each channel, with F1 highlighted being localized in the vicinity of the stimulation site. The response shows several well-delineated components, and the morphology is consistent with those described in literature in areas overlapping with our area of stimulation. And here is a better topographical presentation. And here is one from a patient. So the morphology is different and there are fewer components. Here is a summary of the responses for all the 3 patients. As before, the butterfly plots show the average response for each channel between -100 and 400 milliseconds post-stimulus. In the second panel, the global mean field power of the TEPS is shown with statistically significant components indicated in orange. The topographical distribution of the average voltages for the components is shown below. Next, we determine the highest frequency power of averaged event-related spectral perturbation from the area close to the stimulation. In this demonstrative healthy control on the left, the mean frequency was 27, in keeping with reports from previous literature using similar measures. In contrast, for the 3 patients, the frequency ranged from 8 to 10. Finally, for 2 of the patients, we had the opportunity to repeat the measurements. In the top panel, the previous butterfly plots and topographical presentations are shown, now with a slightly longer post-stimulus interval of up to 600 milliseconds. Following these initial measurements, both patients were started on cannabidiol with the aim to try to improve seizure control. Unfortunately, this was not successful in reducing seizure frequency in either. They were still taking the cannabidiol at the time of the second session, shown here below. We thought it was interesting that it appears that in both, there's an increased amplitude in these late components compared to the baseline session. And we thought that this is interesting because similar large late components have been previously reported in some groups of people with epilepsy and have been hypothesized to perhaps reflect abnormal connectivity. This is ongoing work and also has obvious limitations. First of all, it's a rare condition and patients are challenging to study due to the level of their disability. This reflects the number of trials and the number of sessions that we're able to do, and generally requires a lot of effort from everyone involved. Further, patients are typically on several anti-epileptic medications, and both the medications and the level of epileptic activity may well influence the results and also cause inter-session variability. A particularly important issue is controlling for artifacts, especially peripheral evoked potentials. And this is particularly challenging in this group because patients have difficulties in understanding complex instructions as well as in expressing themselves verbally. Finally, the neuronavigation, including lack of patient-specific MRIs, is a limitation. So in conclusion, Dravet syndrome is among the most well-characterized severe genetic epilepsy syndromes and very interesting from a neurophysiological perspective. In keeping with our hypothesis, our initial data suggest a reduction in the number of early components of TEPS. Further, power spectral analysis showed a lower predominant frequency power compared to control data. And this could suggest altered function of cortical thalamocortical circuits. Whether these changes are replicable and how they are influenced by medication clearly requires further work. If confirmed, it would be really important to characterize whether these changes might be driven by the genetic abnormality or by the phenotype, and as such, whether they might be employed to promote personalized medicine. Finally, I'd like to thank everyone from our incredible team, as well as patients and their families for the significant effort they put in taking part. We're also very grateful to our collaborators, especially for the input and support from Professor Massimini's group. Thank you very much for your attention.
Hello, my name is Mira, and now we do some break exercise together. So take a space and just relax and enjoy. So first we do some lunge. So take a step like this, and you can raise your arms up or up. Yes, very good. Up, up, and one more time. Up. All right. Yes, and then rotate your upper body from side to side. At the same time, you can bend on your knees. Remember to breathe deeply. Up, up, and one more time. Up. Yes, you can shake your arms. Then rotate your hips. A big circle. Up, up. Yes, and then the other side. Yes, good. Oh, all right. Then rise up on your ball of the foot. Whoop. At the same time, you can lift your shoulders up on your ears. Whoop. 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 Yes, and one more time. Whoop. Yes, very good. And the last one, stretch your back like this and open your thorax. Stretch your back and open your thorax. Whoop. Yes, very good. One more time. Whoop. And open your thorax. Alright, you can shake your legs and arms. Up. And now you are ready to continue your day. Have a nice day. Bye.
Hello, everyone, and thank you for taking the time to listen to my presentation today. I hope that you are all staying well in the part of the world you're tuning in from. I'm very glad to have this extraordinary opportunity to describe our research applying biophysically principled modeling of EEG to guide the interpretation and design of non-invasive brain stimulation. And I'm going to be focusing today on a software we've been developing for this purpose called Human Neocortical Neurosolver. So I'd like to begin by describing the challenge in human neuroscience that we aim to address. We all know that EEG is very powerful technology because it gives us a direct readout of human information processing with millisecond resolution. But a downside is what we're recording is the so-called macro scale activity that's generated by large ensembles of neurons. And it's still difficult to infer what's going on at the underlying cellular and circuit level. And this circuit level understanding is really critical if we want to use EEG to help us to understand what non-invasive brain stimulation is doing to neural circuits, and ultimately to help us design better stimulation protocols. Now, invasive recordings are ideal for dissecting circuit level activity, but there's obvious challenges in doing that in humans. So how do we translate from something we measure outside of the head to what's going on at the microcircuit inside of the head? Well, that's the perfect job for computational neuromodeling, where we simulate the electrical activity of the neurons. And with models, we can have specificity both at this microcircuit generation level and at this macro scale recording level. And this is the primary focus of my research program. We've recently turned our modeling framework into this new software tool that we've designed for clinicians and researchers to begin to develop and test hypotheses on the circuit origin of their human EEG or MEG data. And we're calling it Human Neocortical Neurosolver, or HNN for short. And so in my talk today, I'm going to begin by giving you some of the background on the development of this new neuromodeling tool. And I'm going to go through an example of how it can be applied to study one of the most commonly measured EEG signals, a sensory event-related potential. And I'm going to be focusing on a study from my own lab where we've been studying tactile perception and looking at tactile evoked responses. I'm then going to give some examples of how HNN can be applied to study the impact of non-invasive brain stimulation on neural circuits and behavior. And again, I'm going to focus on studies from the lab where we've been attempting to modulate tactile perception using transcranial alternating current stimulation and most recently transcranial magnetic stimulation. So how do we begin to develop a model that can connect these extracranially measured EEG signals to the underlying cellular and circuit level activity? Well, there's two steps along the way. The first is that we have to realize that EEG signals are generated by big electrical currents in the brain. And the first step in getting at the underlying cellular level generation is to perform inverse solution methods that estimate the location, the direction, and the time course of these big electrical currents that we call primary currents that contribute to the recorded data. From there, we need to connect these currents to how they're generated at the underlying cellular and network level. And it's believed that these primary currents are generated by the postsynaptic intracellular current flow in these long and spatially aligned cortical pyramidal neuron dendrites. These dendrites create a big electrical current that creates an electric and magnetic field that's recorded outside of the head with EEG and MEG. And again, it's the spatial alignment and synchronous activity in large populations of these pyramidal neurons that creates a signal that we record outside of the head. And so, given that this is where the signal comes from, there are some key canonical features of cortical circuitry that we have to take into account in our modeling in order to study the cellular and circuit origin of this signal. The first is that the cortex is a layered structure. There's excitatory and inhibitory neurons in supragranular and infragranular layers that are synaptically coupled. including these pyramidal neurons in supragranular and infragranular layers that have these long apical dendrites that extend through the layers. These networks don't sit in isolation, but they're constantly receiving input from other parts of the brain. And there are two primary pathways of information flow into the cortex. One pathway, known as feed-forward input, represents the input that brings the sensory information from the periphery through the thalamus and into the cortical layers. The other type of input is what we refer to as feedback input that comes from higher order cortex or non-lemniscal thalamic nuclei and contacts directly into the supragranular layers. And then again, the primary current dipole comes from the net intracellular current flow across all of the population of pyramidal neurons. And now with this level of detail in a model, the unit of measure of the primary current is current over distance. And this is the same unit of measure that we get out of our source localized EEG or MEG data. And so we would have one-to-one comparison between the output of the model and our recorded data. Now, we know something about the size of the networks that contribute to some of the most commonly measured signals, including event-related potentials. These are typically on the order of 10 to 100 nanoampere meters. They're believed to be generated by the synchronous activity of on the order of tens of thousands of synchronously firing pyramidal neurons. Low frequency oscillations, alpha rhythms, beta rhythms, gamma rhythms, they tend to be larger in amplitude on the order of 100 to 1,000 nanoampere meters, and they're believed to be generated by the subthreshold activity of on the order of 1 million pyramidal neurons. And so this is the scale of neuromodeling that we built into HNN. And next I want to briefly give you some more of the model detail. So we simulate the pyramidal neurons in this model with multiple compartments. And we have single compartment inhibitory neurons, and they're synaptically coupled to each other through both GABAergic and glutamatergic synapses. We calculate the electrical activity in each compartment of every neuron with Hodgkin-Huxley dynamics, where we have active ionic conductances, sodium, potassium, calcium, in the somatic and dendritic compartments. We calculate this longitudinal current using cable theory. And then again, the primary current dipole signal comes from the net intracellular current flow across all of the population of pyramidal neurons. And as I've mentioned, it's important to consider that these circuits don't sit in isolation, but they're constantly receiving input from other parts of the brain. And we have these two pathways of input, a feed-forward pathway and a feedback pathway. Now, in our model, we don't simulate the activity of all these other brain areas. Instead, we generate trains of action potentials that represent the input from these other brain areas. So for example, to consider feed-forward drive, we could simulate a train of action potentials that activate excitatory synapses in the local network in this feed-forward or projection pattern that we call proximal drive, because it effectively hits the proximal dendrites of these pyramidal neurons. This type of input would push current flow up the dendrites to create a positive deflection in our signal. Similarly, we could simulate feedback input as a predefined pattern of action potentials that activate excitatory synapses at the distal dendrites. These push current flow down the dendrites to create a negative deflecting signal. And it's a combination of this feed-forward and feedback drive in combination with the local circuit activity that creates this signal that represents our primary current dipole. Now this is a reduced representation of the network at HNN. The full network contains a scalable number of neurons, and it represents a patch of cortex from a localized brain area. And we've been specifically designing HNN to study some of the most commonly measured signals, including ERPs and low frequency brain rhythms. Now this is a very complicated large-scale network model. All of the code is open source and it's available on GitHub. However, if you don't have experience building and working with neural models, the code can be very difficult to understand. And so in order to make this model accessible to a broad community, we've embedded it in a user-friendly graphical user interface. And so there's no coding necessary to work with this model, where we've created a website that contains installation instructions, background information, tutorials on how to begin to simulate ERPs and low frequency oscillations, and example data and parameter sets for you to work with. Now, when you start HNN, this is the graphical user interface that you see. In the top has a schematic representation of the local cortical column model, and it includes these layer-specific pathways for which you can drive the network to represent your simulation experiment. And so to demonstrate how to use HNN in practice, I'm going to go through an example from our lab where we've been using the model to study somatosensory perception in tactile evoked responses. And so in my lab, we perform tactile detection experiments where we give a subject a subtle brief tap to the middle fingertip, and we ask them, did you feel it or didn't you feel it? We record the brain activity with either MEG or EEG, and we isolate the contribution to the signal from the hand area of S1. And so here I'm going to show you in the HNN GUI what the S1 evoked response looks like from a tap to the finger. And in this example, I'm showing you the response from a fairly strong tap to the finger where the subject reports feeling it 100% of the time. And what you can see is there's a reliable, strong evoked response here with alternating positive and negative deflecting peak amplitudes. And so the first application of our model was to use it to understand how this event-related potential is generated by the underlying neural circuit. And so now I'm going to walk you through how we did this. Now, HNN won't automatically tell you how to generate an evoked potential. Instead, we think of HNN as a hypothesis development and testing tool. And so the first thing we did is we went to the literature and we said, what do we know about how sensory evoked potentials are generated? And it turns out there's a long history of literature looking at the origin of evoked related potentials, particularly in somatosensory cortex. And what that literature suggests is that the S1 evoked response is generated by a sequence of activation to the local network that consists of a feed-forward input, followed by a feedback input, followed by a later reemergent feed-forward input. And so in our model, we wanted to test the hypothesis that a sequence of activation similar to what we knew from the literature could in fact generate this macro scale S1 ERP. And so what I'm showing you here in the graphical user interface is a histogram of the driving spikes that we use in our network to represent proximal and distal drive. And so here we have a histogram of feed-forward drive, followed by feedback drive, followed by this late re-emergent feed-forward drive. We activate the network with these inputs by hitting run simulation, and we look at the output of the model, and we see indeed that this sequence of input was able to reproduce the tactile evoked response. And now, importantly, while the literature gave us information on how to begin the simulation, we spent a lot of time adjusting the timing and the strength of these inputs in order to accurately reflect that recorded ERP. And now, with this simulation in hand, we could go in and look at what happens under different experimental conditions. In our initial study, we looked at the evoked response from taps to the finger that were felt and not felt, and tried to infer what are the underlying circuit differences in these different experimental conditions. What I want to describe to you next is how we've used this modeling framework to understand differences in the evoked response that occur from non-invasive brain stimulation. And so to do that, I'm going to now switch to describe some studies in my lab where we've been attempting to modulate tactile perception with non-invasive brain stimulation. And so I'd like to begin with some background motivation for our non-invasive brain stimulation experiments. So in addition to looking at properties of the evoked response. We've been investigating how brain rhythms impact somatosensory perception. And so what I'm showing you here is during the same tactile detection experiment, we're looking at the spontaneous signal from the hand area of S1 before we tap the finger. And what you can see is that there's large amplitude, low frequency oscillation. When we put a frequency filter on this oscillation using a standard wavelet analysis, you see that this oscillation has a strong component in the 7 to 14 hertz alpha band and in the 15 to 29 hertz beta band. And one of the questions that we were interested in is, does the power of this oscillation in the pre-stimulus period before we tap the finger impact the subject's ability to feel that tap to the finger? And it turned out the answer to that question was yes. And so here I'm showing you that frequency versus time representation sorted over trials where the subject felt the tap to the finger and trials where they reported they didn't feel the tap to the finger. And you can see very clearly that on trials where they didn't feel the tap to the finger, we had higher power in these alpha and beta bands. And in this way, we think of high power as a signature of inhibited perception. And if we do some statistics and we look at the average, so here we're averaging in the pre-stimulus period across the alpha and the beta frequency bands. We're sorting power from low to high, and we're looking at the detection rate as a percent change in hit rate, as a percent change in hit rate from the mean. So the higher it is, the more likely the subject was to feel the tap to the finger. And you can see very clearly there's a linear relationship between power and perception. The higher the power, the less likely the subject is to feel the tap to the finger. And so now, knowing that these spontaneous oscillations are going to inhibit the subject's ability to feel that tap, we next wanted to ask the question, can we use non-invasive brain stimulation in order to enhance these ongoing oscillations, and in doing so, enhance the inhibition of perception? And so to do this, we're going to ask the question, can we modulate rhythms with non-invasive brain stimulation? And if so, can we use H&N to interpret the circuit level effects of the brain stimulation? And we started by looking at these alpha oscillations. There's a lot of examples in the literature that suggest that maybe we can entrain or enhance alpha activity in the brain and influence behavior. And so the first study that we did, we used alpha frequency transcranial alternating current stimulation. We did our tactile detection experiment before, during, and after 10 minutes of individualized alpha frequency stimulation over the primary somatosensory cortex. The stimulation was on for 6 seconds and then off for 6 seconds. And we compared that to a sham condition where we had just one cycle of a 1 hertz oscillation that ramped up and then turned off. And we looked at the EEG signal above somatosensory cortex before, during, and after this TAC stimulation. It turned out that for various technical reasons, we couldn't look at the EEG during the TAC stimulation. And so what I'm going to show you next is a comparison of this pre-alpha stimulation compared to post-alpha stimulation, where we looked at the question, can we enhance alpha activity in our EEG signal? And does that correspond to an inhibition of somatosensory perception? And it turned out with this experimental paradigm, we did not find a significant impact of alpha frequency current stimulation on alpha oscillations in somatosensory cortex. And so here I'm showing you alpha power in the sham and alpha stimulation condition pre compared to post. And in fact, there's no difference in the alpha oscillation. Next, we looked at perception, and we also didn't find a lasting impact of alpha frequency stimulation on tactile detection performance. And so here again, I'm showing you the percent change in perception from the baseline pre-tax condition and the sham and the alpha condition. And what we found was that during the transcranial alternating current stimulation, we did in fact see a reduction in perception compared to sham. However, this reduction in perception didn't persist into the post-stimulation period. And so we didn't have a lasting effect. Now, there were various reasons why our experimental paradigm may not have entrained alpha oscillations or impact perception. We were using a very weak electrical current. We had this very brief on and off, and we had a lot of technical challenges with our experiment that are described in our publication on this work. But we did find one subtle but significant difference in our EEG signal, and that was in the tactile evoked response. If we compared the evoked response above somatosensory cortex in the pre versus post alpha stimulation condition, we found that this peak near 70 milliseconds was greater post-stimulation. And in fact, that was a significant difference if we look at the mean response between 70 and 80 milliseconds. And so this was very interesting to us. And we wanted to see, well, can we use our HNN software to interpret what it is that this alpha frequency stimulation did to the neural circuit to create this difference in the tactile evoked response. And so now I'm going to turn to HNN. Here again is the graphical user interface. I've loaded into this interface the pre-stimulation evoked response, here shown in blue, and here is our actual recorded data. This work was done by a graduate student in the group, Danielle Sliva. So I want to thank Danielle for providing this information. And so now we wanted to test a hypothesis about what this alpha frequency stimulation might be doing to the neural circuit. And we started with the hypothesis that maybe what stimulation is doing, while it's not entraining alpha activity, maybe it's impacting the local synaptic efficacy of the connections within the local network. And so we've created an easy way in the software to modulate the gain of synaptic activity between the neurons. And so you can go in to test the hypothesis that this alpha stimulation changes the local synaptic plasticity. And so to do that, you go into this set parameters button. We have a button here that represents the synaptic gains or the strength between the connections, and you can change them by a multiplicative factor. And so the first thing we did is we said, well, maybe what's happening is that all of the synapses are being strengthened by a factor of 2. And so you can go in and you can multiply all the connection strengths by a factor of 2. This is for the excitatory to excitatory, excitatory to inhibitory, or the inhibitory connections. When we do that, and we hit run simulation and look at the output, we see in fact that an increase in the synaptic efficacy of all of the connections is able to reproduce this difference that we observed in our data. However, it also creates this later difference that we don't observe in our recorded data. And so we have hints that yes, maybe indeed the alpha stimulation is changing the synaptic plasticity. But next we wanted to see, maybe it's not all of the synapses. Perhaps it's only the excitatory synapses or only the inhibitory synapses. And so we went in next and we checked, well, what if we change only the excitatory synapses and increase the strength of those twofold? And what we found was, in fact, this went in the opposite direction. And we got a higher amplitude response, and it didn't match our data. Then we went in and we tested, well, what if it's only the inhibitory synapses? And so here we're increasing the inhibitory synapses by 2 times their strength. And indeed, that inhibitory synapse change was able to reproduce not only this peak difference that we saw near 70 milliseconds, but also the later response where we saw a lower amplitude throughout this post-stimulus period. And so that suggested to us, the model is suggesting that what this alpha frequency stimulation is doing is it's creating a subtle change in the synaptic efficacy from the inhibitory neurons, and specifically only the inhibitory neurons. Now, excitingly, this is actually supported by some of the literature, which suggests that low intensity alternating current stimulation preferentially potentiates inhibitory synapses. And so now we have some evidence from our model that that may be what's underlying the difference in the EEG that we observed. Now, I've been showing you the macro scale current dipole signal, but one of the powerful things in modeling is that we can go in and we can look at all of the microcircuit activity. And so we've created a way in HNN to go in and to view the microcircuit details. So if you go under view, you can look at layer-specific dipoles, you can look at spiking, you can look at frequency responses, somatic voltages, and different things in the network. So here I'm going to show you the spiking activity in the network. And so here's the spiking activity as a raster plot in all of the different cell types in our network. Before we change the synaptic plasticity, representing our pre-tax condition, and once we increase the inhibition twofold. And what you can see is that we increase the strength of the inhibition, we get less spiking in our pyramidal neuron populations. And this decrease in spiking corresponds to a lower amplitude evoked response. And so now we have some very specific predictions about what's happening in the underlying circuit. And we can then test those predictions with invasive recording or other imaging modalities. And we have some targeted directions to go in and further test what this brain stimulation is doing to the underlying neural circuit. And so this was very exciting for us. But in our hands, what we wanted to do was modulate tactile perception. And this experimental condition did not modulate tactile perception in our hands. And so the next thing we did is we went back to our data. And when we looked more closely at our data, we found that, in fact, the beta component of this oscillation is more prominent in somatosensory cortex. And in our hands, it's a stronger predictor of tactile perception. So next, we set out to see if we could instead modulate the beta component of this rhythm, and in doing so, again, inhibit tactile perception. But before doing that, we took a step back to understand our data a little bit better. What I'm showing you here is average data. We're averaging across time, we're averaging across frequency span, we're averaging across subjects, but the brain isn't averaging. And so next we looked more closely at the unaveraged response. And so what I'm showing you here is 10 examples of the pre-stimulus activity before we give that tap to the finger. And this is the corresponding frequency spectrum. And what you can see as these beta oscillations emerge as these transient events. They typically last less than 150 milliseconds. Now, because in the frequency domain, these signals are always positive, when you average in the frequency domain, you get an accumulation without cancellation. And you get this continuous band of high power activity. And you might automatically think, oh yes, there's this continuous ongoing oscillation that's happening before we tap the finger, when in fact, at any moment in time, you either do or do not have one of these transient beta events. This transient nature of beta activity is a very robust phenomenon. We've been fortunate to be able to look at it also in local field potential recordings from the rodent somatosensory barrel cortex. This is data provided by my collaborator at Brown University, Professor Christopher Moore, and a graduate student in the group, Heian Shin. And in the mice, they're doing very similar tactile detection experiments where they're stimulating the vibrissa. And here we're looking at the spontaneous activity in the somatosensory cortex, again, before the tap to the finger or the tap to the vibrissa. You can see that there's very similar expression of activity. We've quantified various features of these beta events in the pre-stimulus period. We sorted the event number, we looked at the event power, the event duration, the frequency span, and we found remarkable homology across not only species, but these different recording modalities that suggests that this transient nature of beta is a very robust and reliable phenomena. This has now also been observed in motor cortex, striatum. Transient beta activity seems to be very consistent across many different brain areas and paradigms. I point briefly to some code that we have on GitHub where you can begin to start to analyze your signals and measure various features of transient oscillations. So now that we know that these rhythms are continuous, we have to revisit the question, what features drive this relationship between pre-stimulus power and detection? Remember, if we had low average power, then the subject was more likely to feel that tap to the finger. If we had higher power, the subject was less likely to feel that tap to the finger. And so when we think of an oscillation being higher in power, we might automatically think, well, that means the oscillation was higher in amplitude, or there was a bigger network contributing to the response. But now that we know that these oscillations are transient, there's several features that could contribute to a difference in average power. It could be that in one case, so here I'm just showing a schematic representation, frequency over time. In one case, in the pre-stimulus period, you have one event versus two events that are the same amplitude. When we average this, we'd get high power, and over here we'd get low power. Or it could be that in both cases we have one event, but in one case the power of that event is higher, and so when you average, you get higher power in the average signal. Or it could be that the duration is longer, or that the frequency span is extended. Any one of these differences could underlie this difference in power in its impact on perception that we've been seeing. And it turns out that it's actually the number of events in the pre-stimulus period that contributes to higher power in inhibited detection. We also found that the timing of the event before you tap the finger mattered, such that the closer it was to the time of the tap to the finger, the less likely the subject was to feel that tap. And the most influential period was approximately 300 milliseconds before the tap to the finger. And this work has been published. I don't have time to get into further details. We've also used our HNN modeling framework to try to understand the circuit origin of these transient beta events. And the modeling, combined with invasive recordings in both rodents and non-human primates, suggests that these beta events are coming from a strong burst of excitatory input to supragranular layers. And again, I don't have time to go into details, but for further details, you can look at our publications on this work. Okay, so now that we know these rhythms are transient, we have to re-ask the question, how might we apply non-invasive brain stimulation to more accurately account for these transient rhythms? And so what we're doing now is we're trying to adapt a new way of thinking, where we're developing brain stimulation paradigms aimed at mimicking the endogenous expression of these transient rhythms. And in my lab, we're currently switching to TMS, where we have more control over the pattern and the timing of the inputs. And we're developing a protocol and asking the question, can TMS bursts of TMS pulses to somatosensory cortex that mimic this endogenous pattern of beta activity, can they be effective at inhibiting perception? But of course, there's a major issue that we're having to deal with, and that TMS is not like endogenous brain activity, because the device itself drives sensory responses. You can feel the stimulation, you can hear the stimulation, it's driving muscle activation. And we now know through many studies that this can be reflected in the TMS-evoked EEG activity. So there are lots of ways to minimize these confounds, but we don't yet have a way to remove them completely. So instead, what we're doing is we're trying to see if we can use HNN to help us understand this confound. And specifically, we're going to ask the question if HNN can help us understand if and how the sensory component of the TEP, TMS-evoked potential, is reflected in the EEG signal. And so now I'm going to briefly show you some preliminary data from the lab, where we've been recording EEG from bursts of TMS pulses to somatosensory cortex. This work is being led by the graduate student in our group, Danielle Sliva, and also a postdoc, Carmen Cole. They're going to be presenting a poster with more details on this work in a session this afternoon, so please tune into that poster for further details. So in this experiment, we're using MRI-guided TMS to give bursts of TMS pulses to somatosensory cortex, and we're recording the response in an electrode above S1. And so here I'm showing you in our HNN graphical user interface the response to that burst of 3 pulses to somatosensory cortex. And these pulses are separated by 20 milliseconds. And you can see that this creates a very strong and reliable response in the somatosensory cortex. Now here we've masked the noise from the TMS stimulator with earplugs, but the subject is most definitely feeling the sensation of this TMS burst of input. And an important thing to note is that this TEP is remarkably similar to the tactile evoked response in S1 from that tap to the finger that I showed you earlier. So it suggests that at least part of what we're seeing may be reflecting the sensory information from this input from our TMS device. And so recall also that we were able to use HNN to look at the circuit origin of this evoked response by driving the network with a sequence of input. And so next we wanted to ask, can a similar sequence of input reproduce what TMS, including the sensory component of TMS, is doing to reproduce this EEG response that we recorded? And so what we're going to do to test that hypothesis is we're going to simulate a sequence of drive similar to the sequence of tactile input, but here we're going to do it 3 times because we're giving 3 pulses of TMS. And so we have this feed forward, feedback, feed forward input that's very similar to the input that creates the sensory component of a tactile input to the fingertip. And we're going to hit run simulation. And in fact, we see that remarkably, this sensory evoked input can almost perfectly reproduce this TEP that we're seeing from our TMS device. And so that suggests that this evoked response is at least in part reflecting the sensory information. Now, importantly, this earlier component of the evoked response may not be influenced by the sensory information. And so what we're doing ongoing in the lab is looking more closely at the earlier components of this evoked response. And again, for further details on that, please tune in to our poster later this afternoon. And so with that, I'd like to conclude. I've described our new software, HNN, which represents a neocortical column model under thalamocortical and cortical-cortical drive that represents this feed-forward and feedback drive to the input. It simulates the primary electrical currents that underlie EEG and MEG with enough detail to connect the macro-scale recording to the underlying cellular and circuit level activity. HNN is now developed into this graphical user interface. It's a user-friendly software which you can download and get all sorts of information from on our new website. And I hope I've convinced you that HNN can be applied to understand the impact of non-invasive brain stimulation on neural circuits through the corresponding changes in EEG signals and can aid in the development of new brain stimulation protocols. And so with that, I'd like to thank my collaborators. I'm fortunate to have an amazing lab at Brown, and everyone has contributed to this work in a meaningful way. The HNN software is being developed by a large network of researchers at different institutions, including Brown, the Martino Center at Mass General, and Yale University. And I'd also like to thank my collaborators at Brown and the Nathan Klein Institute in Columbia, who have been kind enough to share their animal recording data to begin to test some of the predictions that come out of our modeling software. And so with that, I'll leave you with the conclusions and a reminder to please check out our poster later today. And I thank you again for your time, and I'll be happy to answer any questions in the Zoom session that follows. Thank you.
NTMS is a method which helps to contribute strongly to successful treatment of brain tumors. You can even operate patients who come to you and who were told that they have an inoperable tumor.
This is the only way to allocate and locate function in an accurate fashion. It helps me avoiding fiber tracts where function is, either speech or motor function, and I know exactly where the tumor is in location to that.
Now we know the data already before surgery, which means we can involve the patient strongly in these decisions.
Patients are a lot more confident than when they go into surgery.
Patients, the motor mapping is comfortable because they don't feel any pain. We stimulate the motor cortex and try to get some muscle responses and can see this in the EMG. At this way we are able to localize the motor function. The aim of the language mapping is to find out which regions are crucial for speaking.
Helmet. Needle. Cake.
The stimulation leads to a short-term dysfunction. The patient is not able to name the object on the screen.
Box. Leaf. Heart.
NTMS makes surgery safer, resection rates are higher, and in the end the outcome is better. This has been proven by multiple publications of several groups around the globe.
So in one study we operated more than 90% of these patients, and 72% of these tumors were resected without residual. There is a term in neurosurgery that surgery is like a robbery. You need to get the plan to get in and out, and also plan B. And TMS data helps you very much to find this way.
Hi, I am Matti Stenros from Aalto University, Finland. In my talk, you will learn about a common framework for modeling electroencephalography and transcranial magnetic stimulation. Electroencephalography, EEG, measures the bioelectric field generated by the brain. Each EEG lead picks up signal from quite a large region of the brain. This is often called signal mixing. Here you can see how two neighboring electrodes pick brain signals. To unmix the signals and to interpret EEG in the brain frame of reference, we perform source analysis. In all source analysis techniques, the measured signals are projected on modeled topographies of elementary cortical sources. The topographies are computed using an EEG forward model. Here you can see example topographies of two nearby sources. In transcranial magnetic stimulation, TMS, a brief current pulse in a coil outside the head causes a time-varying magnetic field which induces an electric field in the brain, and this E field stimulates the cortex. To estimate where and how strongly we are stimulating, we use a TMS forward model. Here you can see a visualization of the E field inside the skull under a figure of 8 coil. In TMS-EEG, we use TMS and EEG together to study and potentially to treat the brain. In basic TMS-EEG setting, we stimulate the brain using TMS and measure the response using EEG. We can also use EEG features for timing the stimulation and potentially for estimating where to stimulate. Forward models are used in TMS targeting and dosing and in EEG analysis. The conductivity structure of the head has a major effect on the electric field. The physics of interaction between the E field and changes of conductivity is the same for EEG and TMS. When building a forward model, most of the effort is related to modeling this so-called volume conductor. The level of detail of the used volume conductor model varies a lot. For EEG analysis, we commonly use a tree-shell model. Such a model can be constructed using, for example, field trip Toolbox. The TMS community has traditionally used a spherically symmetric head model, but recently the use of highly detailed TMS models has become common in offline planning and analysis of TMS. Here I'd like to highlight the SimNIPS toolbox developed by Axel Thielser and his group. The geometry of the models you see in this talk is extracted from a SimNIPS sample dataset. In TMS-EEG, the TMS and EEG models have been constructed separately, using different segmentations, levels of detail, and solvers. For example, it is common to target stimulation using a simple geometrical projection or a spherical model, and analyze the EEG with a realistic tree-shell model. Or, with today's common tools, one could model TMS using a high-detail model, but analyze EEG using a tree-shell model. If one goes through the burden of making a high-detail volume conductor model, one could use that model for both TMS and EEG. In the rest of the talk, I will illustrate how to do that. In an EEG model, the idea is to compute signal topographies for elementary sources in cortex. These sources are represented using current dipoles. Here is a y-directed dipole in location R1, and the surface potential it would cause in an infinite homogeneous conductor. We'll call this the primary potential phi p. The volume conductor effects are brought in by a linear operator T, and as a result we have potential on the conductivity boundaries of the head. This computation is repeated for all source locations and orthogonal orientations. The operator T needs to be computed only once. For a high-detail model, building T and computing potentials takes about 1 hour with my tools. Now place a tiny coil in location RC outside the head. With time-varying current, it produces electric field in the head. We divide this field to primary and secondary E. The primary component is the field induced by the coil in vacuum, and the secondary component is caused by the volume conductor. We see that the secondary E at R1 is computed with the help of phi 1. In other words, the EEG forward solution of a dipole in R1 is used for computing the TMS-induced E field in R1. Compared to solving the potential, all other computations are fast. In the previous slide, I illustrated how to compute the surface potential of one dipole and, using a tiny coil, one component of the E field in the corresponding location in the cortex. To make a full E field model, we just repeat the computation for cortical locations of interest and represent the realistic coil as a combination of these tiny coils. All potentials phi can be pre-computed. For each coil location, we only need to calculate coil-related integrals. This can be done very fast, even in real time. If you want to learn more about this, please see our publications. In summary, in TMS, forward models are used for offline planning and analysis and real-time navigation and dosing. In EEG, they are used for source-level analysis. The physics of how the head interacts with the E field is the same for TMS and MEG, and the EEG forward model can be formulated as a part of TMS forward model. In TMS forward modeling, the EEG part is the laborious one, but it is independent of TMS coil parameters and can be pre-computed offline. For TMS-EEG, a common modeling framework like the one presented here should improve the quality of experimentally used TMS-EEG models while saving effort. Thank you for listening.
Everybody, this is strange, isn't it? I'm very sorry not to be able to join you all in Finland, but I just want to say thank you for tuning in for this talk on cortical depth across the brain surface. So why are we doing this? Well, I guess we're all here at this meeting because we believe in some way that brain stimulation, whether we're talking about TMS or one of the electrical modalities, has some role to play in helping people to do things better. So whether we're talking about fixing neurological problems, or maybe we're talking about cognitive enhancement in otherwise healthy people. But we have some challenges, and I guess the biggest set of challenges really are about targeting and setting dosage for brain stimulation. And there are lots of different ways of thinking about that, but effectively we're talking about the ability to decide where and when to deliver our stimulation and how much of it we should give. One problem in dose setting is that we don't really have a good model of how electrical or magnetic energy affects the brain, or at least gets from the stimulator to brain tissue and thereby has an effect. Now, we know, I guess, the physics behind all of this, so we know there are things like Maxwell equations and Lorentz laws and things. And when we know some of these principles, we can combine them into computational modeling techniques. So you'll see many beautiful images like these finite element models that are produced. I'm sorry for not crediting this. This is from Andy Huang's roast toolbox for MATLAB. One thing that we that adds variance to our models is cortical depth. So why does that matter? Well, we know that at least for TMS experiments, the amount of extra output you need from the stimulator to have the same effect, given a difference in cortical depth, is about 3% per millimeter. So if the brain area that you're talking about or trying to target is 1 millimeter deeper than some reference level, then you need to turn your dial up by an extra 3%. So there's some clever work over the last few years by Mark Stokes and colleagues that helped us to understand the role of depth itself, so irrespective of the intervening tissues. My interest here is actually in the variance of cortical depth. And in fact, not just variance, but the coefficient of variation. So the coefficient of variation is the standard deviation divided by the mean, and it's scale free. So for most distributions, the standard deviation or the variance should increase as the mean increases. That's not true for for coefficient of variation. So that allows us to take out the effect of different depths of cortex and to replace that with a scale-free measure. And why would we do this? It's because if we find a brain area that has relatively low variance or low coefficient of variation, we can use reasonably simple methods for targeting that area or for dose setting for that area. Whereas if a brain area has relatively high coefficient of variance, so if between participants it's if it's shallow in some people and deep in other people, then we have a problem and we need to use more specific methods to target that particular brain area. Okay, how have we done this? So this uses some pre-existing data. In fact, these were taken from the OASIS database, which is freely available. There are something over 400 images in the OASIS 1 series. I was only interested in people in their sort of middle years, so 25 to 60 years old, where the structure of the brain is relatively stable, and people with a dementia score of 0. The full methodology is described by Marcus and colleagues in a paper that's cited here. But we have 94 images in this database, so that's the number of images that contribute to what I'm about to show you. Okay, image processing. Right, there are a couple of steps here. So firstly, SPM12B was used to segment the images into the two parts of the image that are of most interest, and those are the grey matter, so the place where we assume TMS and TESS are having their effect, but also the soft tissue. So the soft tissue layer in SPM segmentation includes the scalp. To find the scalp surface itself, we can just apply a 3-dimensional convex hull. So it just fits the minimum frame that fits the outside of the soft tissue layer. So that's a proxy for the scalp layer. And then from there, for each point in the grey matter image, or the grey matter segment, we can find the minimum Euclidean distance from that point to a point on the scalp surface. So where is the closest point on the scalp to that brain region? So we do that for each person. We can then take those distance images and transform them into a standardized space, in this case, MNI space. And having everything in a standardized space means that we can do group statistics on everybody's image all in one go. So we can, in this presentation, I'm only going to talk about the coefficient of variation and some correlations. Okay, so what do we find? Well, one thing we would look for in our group data is whether the group data matches what we expect a cortex to look like. This is an inflated image of the brain. So, so we've taken the mean of all of the MNI format images and just put them all together and found the mean depth of each of those voxels. Fortunately, it looks roughly the way we would expect. So, so a gyrus, which is relatively close to the brain surface, to the scalp surface, should be in a relatively bluish color, so a smaller number, whereas deeper areas, particularly folded underneath the brain, under the temporal lobe, we would expect those to be deeper and a redder color. So that's reassuring. Just to orient people who aren't used to looking at inflated brains, the central sulcus is indicated here, and it should be relatively deep compared to the precentral and the postcentral gyrus either side. Now, if we talk about the standard deviation of depth, now the pattern isn't quite so obviously mapped onto cortical folding. Why would that be? It means that there's some variance introduced into this measure that is not related primarily to depth. And the way to spot that is to divide the standard deviation image shown here by the mean image shown in the previous slide to get this image, which is the coefficient of variation. Now, the coefficient of variation suddenly has a much wider distribution across this brain surface. And this reflects two things. So firstly, there's a process variance, so the variance in warping each individual brain image into a standardized space. So SBM works very hard and is very good, but there's going to be some variance introduced there. But also there's some source variance, so variance in the folding of different people's cortex. And I think between these two images, there are ways to tease these apart, but these are the sources of variance that we would need to worry about in talking about standardized brain spaces. Okay. Now, just to make things a little bit simpler, because that's a complex image, we can look at regions of interest. And so if we take some commonly stimulated regions of interest, so there's a middle frontal gyrus here, there's a precentral gyrus, angular gyrus, the superior occipital cortex gyrus, and superior temporal gyrus. These are brain areas that contain interesting functional areas, and so are commonly used in brain stimulation experiments. So looking at the coefficient of variation of these regions, well, there's some variance across those areas. So, for example, the occipital cortex has a relatively high coefficient of variance, and also seems to be different between the left and right hemispheres. However, relatively low variance in the prefrontal cortex, and that's a bit more optimistic for example, cognitive experiments where we want to knock out or enhance some kind of cognitive processing. Or this is a common target for depression-related targeting as well. Now, thinking about correlations in depth, well, one thing that will be a good sanity check is whether brain or head size correlates with age. The size of the head does not correlate with age, so your head does not shrink or grow as you get older. But your brain volume does. In fact, it declines in volume at about 1 milliliter per year, terrifyingly. What does that mean? Effectively, that means that your brain is shrinking away from your skull. So you would need to turn your stimulator up a little bit for each additional year of age. But there's another way to think about correlation, and that's to take places that we usually use to set our dosage, for example, primary motor cortex, and see if the surface of the cortex correlates in depth with that region of interest. So taking the primary motor cortex, adding a little seed region here at the top, does the depth of the cortex correlate well with that, with the depth of that region of interest? The answer is not particularly well. So there are some fairly good R values, this is Pearson's R, of about 0.6 or minus 0.6, so negative correlation is fine too, but they tend to be very closely clustered around the seed region. What about the primary visual cortex? Again, not particularly well. So again, there's clusters of high correlation around that seed region, but away from the seed region, not so good. Okay, so this is a quick tour through some statements about the depth of the cortex across the head. What can we say about this? So one useful sanity check is that the mean depth of the cortex varies in the way we would expect it to. But standard deviation and the coefficient of variation also vary across the cortex. And given that coefficient of variation should be scale-free, it helps us to find areas where depth can be well estimated or poorly estimated. And the answer is actually not very many areas have a good correlation. Oh, sorry, good coefficient of variation. Talking about those correlations then, the depth of the cortex correlates poorly with the seed region in primary motor cortex, so motor evoked potentials, MEPs, are not that good as a predictor of how much energy to deliver to a particular brain region. And that's the same, in fact that's even worse for the primary visual cortex, so setting dosage according to phosphine production is not a reliable way to find the energy suitable for stimulating a nearby brain region. To wrap all this up, so firstly, brain stimulation dosage needs very good prediction of cortical depth. Given that the required dosage changes by this magic value of about 3% per millimeter, we do need to know how deep a particular brain area is. There are other factors, of course, but depth is a clear source of variance. But the way we typically do TMS experiments, which is to find the stimulator output required to generate a motor evoked potential and then set dosage as a percentage of that, actually is not a good way to do it, I think. I think this is what the data here is saying. So motor evoked potentials and visual evoked potentials, or phosphines, are not a good way to set dosage. What that means is that for most applications, we would need a person-specific brain image. So you need to scan your participant and use that in some kind of neuronavigation system to understand exactly how to target a particular brain region with a particular amount of stimulator output. Why are we bothering to do this? Well, so where could we go with this? Well, in the analysis I've shown here, I've only taken people in the sort of middle years of their lives, so 25 to 60 years old, where brain structure is relatively stable. But we can look at people who are older, so if we can see this image properly, so we have people in their middle years have a sort of nicely packed sort of skull, so the brain reaches out to the edges. Whereas in an older person, and by that I mean people in their 90s, the brain is starting to move away from the outer surface, and the ventricles are also increasing, so the amount of fluid in the head is a bit larger. So understanding brain stimulation targeting in aging is a key area, and also in other applications such as eating disorders. So a person with an eating disorder typically has a very different brain structure to others. And of course, another application would be to verify TMS models. So can we use the variance estimate of say, primary visual cortex to map onto the known variance in the stimulator output required to generate visual evoked potentials? So can we map these predictions onto real data? Okay. That was a very quick tour of some depth data. I'd be very happy to chat about this. This is extremely exciting, being able to do this online. So please do join in, or please do get in touch if you're interested. My email and Twitter handle and things are down there. But meanwhile, thank you very much.
Hello everybody. I will present today our preliminary results of the study Gender Effects in a Subsample of the Human Connectome cohort. The study is based on simulation, and my background is the research group Neuromodulation and Multimodal Neuroimaging. It's located at the University Hospital LMU in Munich, Germany. We've done in the past a lot of different multimodal neuroimaging studies. Today we will focus only on the modality of T1 weighted and T2 space data from the Human Connectome project dataset. This data is freely available, and we do the simulations exactly as we have done in the past, with 2 milliamperes, and the electrodes will be located on the left DLPSC and the right DLPSC. There is anatomically variability. If you are looking into the cycle deaths, that's a study by Hill and colleagues in the Journal of Neuroscience 2010, then you will see differences that go up to a factor of about 7 in the adult brain, and even in the infant brain, we are here in a factor of about 4. And this variability is even found in monozygotic twins. And when we are looking into one of the largest data sets so far in the Enigma cohort study from Kong and colleagues, then we see that sex is strongly influencing their cortical thickness in areas also including huge areas of the frontal brain, and also the brain size is completely influencing cortical thickness. We are applying on this variable brains non-invasive brain stimulation. Our focus in our study was to investigate gender and brain size effects in regard to simulation on a larger cohort of healthy subjects. The sample we used is based on 450 subjects in the age range of 22 to 36. There are a gender distribution of about 250 females and about 200 males. The structural data has a spatial resolution of 700 micron isotropic T1 weighted and T2 weighted images that were all acquired by the Human Connectome project. There are detailed demographic and behavioral data and also multimodal MRI. There are 210 non-twins, 130 monozygotic twins, and 106 dizygotic twins. We have used SynNIPS E-field computation using the Salad Atlas ROI and a proxy of it. The Salad ROI is based on frontal cortex atlas estimation by resting state and DTI in humans and macaques. The proxy we have applied is based on the MNI coordinate in a sphere of 18 millimeters. It's shown here in a blue binary coding, and here it's overlaid on the solid areas in those axial slices. The method of SynNIPS simulation has been published by Windhoff, Alexander Opitz, and Axel Tischer in 2013, and it's based on T1 weighted data. You could also include T2 weighted data. They use FSL and FreeServer, and they take into account skin, skull, eyes, and tissue groups like gray matter, white matter, and CSF. They are all different weighted, and when you do simulations On the motor cortex, you see that with increased E field, with increased stimulation strength, you get higher E fields. Also, like the stimulation side matters, so meaning if you locate the electrodes more anterior or more posterior, you will have a different distribution of the E field. We have used FreeServer 6.01 with LERM at the LLZ Supercomputing Center in Munich. Thereafter, we used the SIMNIPs based on our own memory scripts on a local cluster. The results. We found a remarkable effect of gender on the E fields in the DLPFC region of interest. Their effect sizes are moderate, but the sample is really large with 450 subjects. Their E fields, the mean E fields, are associated with an increased intracranial volume that has been found to be larger in males than in females. We did not find any age-related effects on the DLPFC ROI in this cohort, but this could also be related to the fact that their age range is in a younger age. If you're looking here more specific into the different percentiles calculated by SIMNIPs, you will find a strong effect of their intracranial volume, here shown in the form of different distributions for their gender, and also are quite different E field results based on gender. Here shown for their 95 percentile, 99 percentile, and 99.5 percentile for the norm E fields. There were no gender effects. Here in this picture, it's a different display from the just like distribution plots, and here you also see that for males, it's for all 3 percentiles quite larger. When we go into more robust statistics like Bayesian statistics, we see a strong effect starting with a sample size of about 200. We've done in the past one study that has been published by Lucia Bulabas from our research group, where we looked into different solid left DLPFC regions of interest and used the data of the today largest clinical trial on major depression. There we found that increased gray matter in the left DLPFC is associated with an improved antidepressive response. The interesting fact is that this was only specific, this gray matter area, for the modality of prefrontal TDCS. It was not given for the antidepressive medication or the placebo condition, the 2 other arms. Their effect sizes are shown above, and these effects do reflect some preliminary interpretation that we could use DLPFC gray matter for therapy planning. Why this is relevant for psychiatry? If you're looking into this large meta-analysis of 193 voxel-based morphometry studies, it clearly shows up that the bilateral insular cortex and the ACC is affected across different psychiatric disorders such as major depression, schizophrenia, or bipolar disorder. This has also been found for the resting state data. This study has been replicated by Shah and colleagues in biological psychiatry, and they again found these areas. They are also for the resting state are known as the salience network. Interesting. When we are looking back to our studies, starting with our frontal stimulation paper on resting state, from 2011, we found some medial areas where we found the strongest significant effects. You find those effects also in the simulations between those electrodes, but also in the effect for the task-based fMRI and even in the EEG. Here is some meta-analysis on new query on major depression. And again, when you include 896 studies, you find a pattern in the left DLPFC and the ACC that is strongly affected by the disease. We think that with the stimulation, we are on the right location. We found in our study here a significant gender effect on E-field strengths. We found no effect of age in healthy subjects, but we've done previously a study by Mitsutani and Takahashi that is in preparation, where we found that for diseases like major depression, schizophrenia, there is an age effect. So meaning a decline in E-fields depending on their age. That has also there not found for their healthy subjects. Their ICV, the intracranial volume or the brain size, is directly associated with E-field strengths. On this data set, it will be interesting to look into genetics because we have in this cohort monozygotic twins, dizygotic twins. And another interesting factor will be the layer fMRI, as the human connectome cohort also includes 7 Tesla data. The question that remains from this study is, should we use individual-based simulations before we plan our next studies or before we apply stimulation to patients? Thank you very much for organizing this exciting conference. Also, many thanks to all the people contributing to the study. The most of them are also part of the research group Neuromodulation and multimodal neuroimaging. I'm open for the discussion and will just like announce that we will also publish all our scripts on Zenodo. Thank you.
Hi everyone, here is Giacomo Bertazzoli from Italy, and I'm going to show you my project on the impact of artifact removal methods on the TMS-EEG signal. So concurrent TMS-EEG is a combination of transcranial magnetic stimulation and electroencephalography. And it allows to stimulate a small area of the cortex using the TMS and to record how the signal spreads through the cortex with the EEG. Here's an example of an experimental setting of a TMS-EEG experiment. And what you usually get from a TMS-EEG experiment is called a TMS evoked potential, a TEP, which is conceptually equivalent to an ERP. But here the evoking event, the stimulus, is a TMS pulse. It's a direct stimulation of the cortex. This technique is used to investigate brain connectivity, since you can stimulate one area and record how the signal spread to the connected areas with the EEG. So you can make an inferences on which area is connected to the one you stimulated. And this is used in basic research to study brain connectivity, but can be also used in clinical research to develop new biomarker for integrity of brain connection and also to probe the reactivity of the cortex. However, raw TMS-EEG signal is very noisy. This is due to the fact that the TMS pulse interact with the sensible EEG recording, causing TMS-evoked artifacts that you can see, for example, in this raw TMS-EEG signal. You can see, for example, that around 0, there is this huge TMS-evoked artifact, which is the pulse artifact, which actually goes up to some millivolts. And also later, the decay artifacts, muscular artifacts, the TMS-evoked ocular artifacts, and all other artifacts that impair the use of the raw TMS-EEG signal. For this reason, there were developed a lot of offline removal methods to extract from these raw TMS-EEG signal the useful TEPs that in the end look like this nice signal here, which is, as you can see from, for example, from the scale, very different from what we saw before. So this is an important and heavy phase of TMS-EEG signal processing. The point is that there are several methods to do this, but there is no standard method. So we ask the question, how the choice of the preprocessing method is affecting the final outcome, which can be translated in to what extent the choice of a researcher to use one method instead of another one will affect in the end the results. Or it can also be seen as how safely can we compare the results from two different TMS-EEG studies that use different TMS-EEG processing methods. So how do we check it? So we took a larger TMS-EEG dataset from a study that wanted to investigate brain connectivity using TMS-EEG in the dorsolateral prefrontal cortex area and in the inferior parietal lobule. And we processed it with 4 different processing methods. First one is ARTIST, which is an automatic algorithm that inputs the raw TMS-EEG data and output the clean TMS-EEG data. Second one is TMS-EEG, which is a fully manual graphical user interface that guides the researcher through the processing. Third one is TESSA, which is actually a set of functions that a researcher can use to build a processing pipeline. We use the default pipeline that I suggested. And lastly, sound SSPCR, which, as opposed to the other one, it doesn't use any blind source separation algorithm, but it uses sound and SSPCR, which are new sophisticated algorithms that allow to clean the TMS-EEG signal without any ICA. And here the resulting TEPs, for example, in the dorsolateral prefrontal cortex. So we had 4 outputs since we had 4 processing methods. Here for the dorsolateral prefrontal cortex, and here for the inferior parietal lobule. Then to test our hypothesis, we just compared these 4 TEPs to look at differences and how this signal correlated with each other. So here, so the result. I'm going to show you the result just for the inferior parietal lobule for time reason, but also because the results in the dorsolateral prefrontal cortex are very similar. So here are the 4 outputs. And the first thing you wanted to check were differences in the amplitude of the signal. So we looked at point by point, channel by channel differences using the cluster-based analysis in the field trip. And here are the results. Here in the row, you can see the comparison, each comparison. There was 6 comparisons since we had 4 conditions. In each column, you can see different interval in times of the whole epoch we had. In the last column, you see the whole epoch, and each one of that butterfly plot represent the differences in the TEPs. The colored line on the bottom on the butterfly plot here represent positive and negative significant cluster from the cluster-based analysis. On the topographical plot, you see colored the voltage distribution, which is the difference of the 2 conditions, and the white dots represent significant channels, so channel in which we found a significant difference in amplitude. The first thing you can see is that you can spot differences, significant differences in the whole epoch, especially in these first 3 comparisons, which were all the comparisons with artist methods. You can see differences also in the other comparison, but less prominent. So the second thing we wanted to check is how these signals correlated with each other. And the correlation can be seen both in temporal dimension and in spatial dimension. So we started with the spatial dimension, and we looked at the topographical correlation. What does it mean? So we had our four signals, and we compared each instant, which is a topography of the signal, and we checked how these topographies correlated with each other. So it's the topographical correlation. And here are the results. So each colored line here represents one comparison, one correlation. On the y-axis, you can see the amount of correlation, and on the x-axis, you can see the time. These straight lines here represent the significant instant in time in which the correlation, so in which the topographical correlation was significant. And the first thing you can see is that the topographical correlation was significant basically in every instant in time, except from the beginning of the signal. And also you can see that at the beginning of the signal, the amount of correlation is low, while it gets better while in later latencies, but not for all the comparisons. You can notice that for the comparison with artists, with this method artists, they remain low until almost 100 milliseconds. Second correlation we checked was the temporal correlation. So instead of looking at the correlation of the topographies, we look at the correlation of each channel in the whole epoch. So for example, this black channel in all conditions. And we looked how the signal correlated in time. And here's the results. In the first column, you see the correlation of each channel for the whole epoch. Again, the white dots represent significant channel, and color-coded is the amount of correlation. First thing you notice is that all channels are significant, and also that the correlation for the whole epoch is high, basically everywhere. But if we split the correlation in smaller window, we notice that in the early latencies, there are areas in which the correlation is low, which are the frontal areas. Here, you can see here and here. The conclusion, so. The initial question was how the choice of the preprocessing method is affecting the final outcome. And the answer is not straightforward, of course. Different methods showed different level of agreement. We see it in amplitude that some methods like artist showed a lot of differences, while other agreed more, for example, TMS-EEG and sound. We saw in the spatial patterns, like we saw the correlation in topographies was low in the beginning, and it was variable in later latencies. And also for temporal patterns that we saw in the temporal correlations, we saw that for the whole epoch, the correlation was very high, but for initial latencies, the correlation was low, especially in the frontal areas. So the choice of the processing method impacts the final TPs. But the real question is, what is the amount of variability that we accept in this phase of the preprocessing? And also where all this variability comes from. Different methods uses different strategies and also different level of automatism. And in the end, it sums up in a different amount of data removed with the respect of the raw data. For example, if we look at the rank of each dataset after being cleaned with different processing methods, we notice that the rank, which is an estimation of how many channels were interpolated, how many ICA components were removed, we see that artist removes a lot of, interpolates a lot of channels, remove a lot of ICA components compared to the others. While, for example, TMS-EEG is more conservative and it spares most of the signal. So this can be one place where to find an explanation of all this variability. But the main question that remains is, do we need a standard for the processing method, or do we accept all this variability that remains? So I hope I can discuss this question with you in the Zoom discussion room after the talk. So see you there. Goodbye.
Hi, my name is Tuomas Mutanen and I currently work as a postdoctoral researcher in the Department of Neuroscience and Biomedical Engineering, Aalto University, Finland. Over the course of the past few years, I've been developing some spatial filtering methods for tackling some of the very challenging artifacts related to TMS-EEG recordings. These novel methods have now been included as a part of an open source data analysis toolbox called TESSA, and one of the main purposes of this video is to show you in practice how you can take and use these tools to clean your own TMS-EEG datasets. In addition, I will go through some theoretical foundations of these methods to help you understand what they are made for and how do they work. So I will give you a super quick introduction to combined transcranial magnetic stimulation and electroencephalography, because I'm sure that for the vast majority of you, it is a very familiar method. So the idea, of course, is that we bring a coil next to our subject's head, and then through the electromagnetic induction, we induce an electric field in the brain strong enough to activate the neurons in our cortical region of interest. Then by following the brain activity simultaneously with EEG recordings, we can, for instance, follow how this initially focal activity spreads to the other cortical regions, and thus we can learn something about the connectivity. The problem, of course, with the TMS is that the electromagnetic induction also couples the TMS pulse to other tissues than just brain, and for instance the cranial muscles, or it can couple the TMS device with the EEG instrumentation. And as a result, we get a number of challenging artifacts in the TMS-EEG signal that we want to remove. Luckily, we know quite a bit of the underlying physics and physiology of the neuronal EEG signals of interest. We know that the neuronal EEG signals are coming from the postsynaptic currents, mainly on the cortex, and these postsynaptic currents, they drive volume currents that must travel through the insulating skull, and then they travel through the conductive skull. EEG then is measuring the potential differences on the scalp resulting from these full volume currents. Now, because of this conductive structure of the head, we know that the genuine neuronal responses, they generate topographies that are rather smeared and that have rather low spatial resolution. This is not the case for many of the artifact sources. For instance, the TMS evoked muscle artifacts, they can have topographies that have a much higher spatial resolution. So if we can take this prior information and write it in terms of mathematical formalism, we can use it in a powerful way to detect and remove artifacts from our data. This is done here in the equation that we can see. So our signal S can be written in terms of the artifacts A, the noise N, and then the unknown cortical signals X, which are linked to our EEG signal through the lead field matrix, which basically describes how our different EEG sensors are sensitive to all the possible cortical signal sources that we might have. By using this equation in a smart way, we can remove some challenging artifacts from TMS-EEG signal, as we will see today. So the tools we are using today, they work on MATLAB. So obviously the first thing to get started is to find and download the appropriate code. So as I said, the tools are now part of the open access data TMS-EEG analysis code repository called TESSA. And you can find all the relevant information of the TESSA toolbox from this website. And the TESSA website obviously has links to the releases, which you can download, and then a very comprehensive manual that explains you how to download and install the TESSA tool package, and also how to use the functions for different purposes. If you want to contribute to the TESSA toolbox, and then I would strongly recommend to contact Nigel and take it from there. Now the TESSA code repository has been implemented in the EEG lab environment, and EEG Lab is then another MATLAB data analysis toolbox. It's very popular among the EEG analysts, and cool thing about the EEG Lab is that it enables you to download various types of EEG datasets almost regardless of your EEG system, and also it is easy to upload the EEG Lab data that you have pre-processed to, for instance, Fieldtrip, and continue with some more advanced analysis. The link for EEG Lab you can find here. And then a third toolbox that you need to get everything working in TESSA is this fast ICA package, which is basically an efficient algorithm for finding the independent components. And this is the link to fast ICA. Now, after we have successfully downloaded the appropriate toolboxes, we can go to the first method. And the first method we will go through today is so-called signal space projection combined with source-informed reconstruction, in short SSP-SIR. And this method was originally developed for removing the TMS-evoked muscle artifacts in the EEG signal. So on the first slide, you will see how the TMS-evoked muscle artifacts typically look in the EEG signal. So on the left panel, we can see the characteristic biphasic responses right after the TMS pulse, as well as the topographies that are typical for muscle artifacts that have a much higher spatial frequency than, let's say, neuronal topographies. On the other hand, on the right side, we can see the time frequency transformation of a typical muscle artifact contaminated EEG time course. And the goal here is now to estimate those muscle artifact topographies that you can see on the lower left panel. And we estimate them from the high pass filtered data, because we can see clearly from the right side panel that the neuronal signals, neuronal responses are mainly confined below 100 Hz. And the whole sequence of the SSP-SIR method goes as follows. So first, we estimate the muscle artifact topographies from the high-pass filtered data, and we project those topographies out with this projection matrix P. And now you can see that the new equation, the main difference is that we have got rid of the A, but the new equation has the exact same form as the original one. So we can use traditional inverse estimation methods to estimate the cortical sources that will generate the artifact-free signal. And because we have the lead field matrix L and the source estimates now, we can actually go backwards and reproduce the artifact-free signal in the original signal space. So let's then see how this SSP-SIR implemented in TESSA looks in practice in our MATLAB. So what we have here is some example datasets where the left parietal cortex has been stimulated. And now, regardless of some very basic pre-processing steps that I've done, it's clear from these both figures over here that there is still some residual muscle artifact left. So from this figure, we can see that there is this early topography clearly coming from the temporal muscles, even though the parietal cortex was stimulated. And it's a bit subtle, but if we check some of these channels in this global EEG map, we have these very fast biphasic responses in these channels, characteristic to muscle artifacts. So let's see how the SSP-SIR will work. So we will open the SSP-SIR using this graphical user interface of TESSA. And these are the default settings. One important thing to note is that you do need the lead field matrix. I recommend to use a anatomy-based subject-specific lead field if you have one. But if you don't, then TESSA will provide a spherical 3-layer model-based lead field that you can use to clean the data. And it's doing its job. Before we have the final result, we need to decide how many muscle artifact components we are removing. And the SSP function will give us 3 figures from which we can determine this. And here I think all the figures tell the same story. So I will remove 2 components because the first 2 components have this clear broadband response that I talked about being characteristic to muscle artifacts. And similarly, these 2 first components have these sharp initial responses. From the third figure, we can see that the first 2 components explain by far the most power of the artifact signal. And now if we visualize the data, again, we can see that those muscle artifacts have disappeared. Now the cool thing in EEG Lab is that if we write the EEG Lab history, you can see the command that we just used, and you can copy and paste it to your script and use it the next time in your code. And that is exactly what I like to do when I build my own pre-processing scripts. The second method that we are going to go through today is called the source-utilized noise discarding algorithm that we abbreviate to SOUND. The SOUND is based on very similar ideas as the SSP and the similar theoretical concepts that I very quickly went through in the beginning of this presentation. The overall idea of SOUND is presented on this slide. You can read the detailed algorithm from the original article, but the main idea is to evaluate the noise levels of all of our channels through a cross-validation scheme where we utilize minimum norm estimation and then extrapolating the clean signals from the minimum norm estimates. Once we have the final noise estimates for all of our channels, we can form a optimal Wiener filter, a spatial filter, which removes the noise and artifact sources that have topographies that are not likely to originate from intracranial sources. Okay, so let's look at now an example of the SOUND algorithm. So again, we are in my MATLAB, and I have pre-processed this example dataset using some very basic steps. But it's clear that there is still some artifact left. Specifically, there are some bad channels that you can see as these long depolarizations. You can see them both in this butterfly plot on the right clearly, and then also in the left in the global EEG plot. So let's see how well SOUND will perform in automatically finding and removing these artifacts. So again, we can look for the SOUND tool from the graphical user interface. And here the default settings, which usually work quite well. And you can give your own lead field matrix if you have one, or then you can let TESSA to form the theoretical 3-layer spherical model-based lead field. And the SOUND has been speeded up quite substantially since it came out, so nowadays it will only take a few seconds for this SOUND algorithm to clean the data. And we have the result already, so let's see how the plot changed. And quite clearly, the neuronal signals which were underlying, they have stayed intact. We have some nice responses that could definitely be coming from the occipital cortex, which was being stimulated here, but we got rid of the bad channels. And again, we use the graphical user interface, but you can get the code by writing the EEG history. And next time you can clean the data in the exactly same way by adding this command to your script. So that's it. I hope you found this video useful and informative. If you still have some further questions or you need some further assistance to get started with this method, please don't hesitate to contact me by my email. Similarly, if you find any bugs or problems in the code, reporting them to us would be highly appreciated. In the end, I want to thank all of my collaborators, and with bold, I'm highlighting those who especially contributed in making these methods open access.
I will present you something we call the TMS-EMG Clicker, an online platform for physiological data annotation. In order to understand what the platform is and what are the specific challenges we help to overcome, let us look at the typical TMS-EMG experimental setup. TMS, or transcranial magnetic stimulation, is a non-invasive brain stimulation method which allows effective stimulation of the brain regions which are relatively close to the skull and scalp. In TMS, we induce a small current in the brain by creating a rapidly changing current in a wire loop, usually called the TMS coil or the TMS transducer. A changing magnetic field in turn induces an electric field, which drives charge separation inside the brain tissue and can lead to activation of neurons. However, the strength of the field decays with the second power of the distance from the coil to the target area. For this reason, we always get the strongest stimulation near the superficial regions of the brain. When performing TMS experiments, we need an easy way to detect that we have stimulated the brain. Two major requirements arise. The stimulation site in the brain should be relatively superficial, and the results should be a stimulation we can measure in some way. Probably the easiest responses to get from the brain stimulation is a muscle twitch. Activating certain parts of the brain leads to activation in specific muscles. These activations can be observed visually. However, a more quantitative way is to measure the electrical activity in the muscle using electrodes which are attached to the skin near the muscle. This is called electromyography or EMG. An electromyogram tells about the activation state of a muscle with respect to time. It is quite obvious why TMS and EMG are often combined as method. With TMS we can induce muscle activation that is readily measured by the EMG. This provides an easy framework to test hypotheses about the brain functionality and also to test different aspects of brain stimulation when we are developing new methodology. So what is this clicker and why have we developed it? Here are a few quotes which I have extracted from various interviews with researchers. I need a reliable algorithm that detects EMG responses after brain stimulation. I want to know what is the variability between annotating muscle responses, especially the onset time of the response time after the stimulus. I see differences of even a few milliseconds in the onset time of the brain response reported as meaningful. I would like to know how accurate and reliable these measures are. I'm developing a machine learning algorithm for classifying brain responses. I need a large database of human annotated brain responses in order to do this. Our platform aims to facilitate the issues raised in these comments. And how do we do that? I will present 3 hypothetical researchers and show in which way their capabilities and needs can bind together in a synergistic way. Let us start by considering a researcher who has just conducted a TMS-EMG experiment with 10 different subjects and has gathered altogether 500 potential EMG responses from each subject. There are now 5,000 responses to analyze. Some algorithms can already annotate these data automatically. However, our researcher takes their responsibility seriously and only relies on data that can provide a reliable result with a trustworthy estimate of the errors involved. Unfortunately, a proprietary algorithm provided by the software cannot guarantee these properties. Therefore, their only option is to manually go through each piece of data and annotate them by hand. Unfortunately, many of us has noted that there are not many easy-to-use tools for this purpose, and we often end up writing our own tools. With MATLAB, for example, these scripts often end up being used by a single researcher. Our tool, Clicker, provides a platform where these data can be annotated. In our preliminary test, we achieved annotation rate of up to 700 annotations per hour. With improvements and more experienced users, we expect that this number can rise even higher. In the case of our hypothetical researcher, they can upload their data to the Clicker database and annotate them using the tools we provide. I will talk about data security a bit later. Now let us turn our attention to a second hypothetical researcher. They are writing an algorithm for automated analysis of brain stimulation responses. They got a set of few thousand responses from a colleague, which they are using as a dataset. The algorithm could be a deep neural network or an autocorrelative time series analysis. or something else completely. It does not really matter in this context. However, let us assume that they want to write a deep learning algorithm that detects the onset of a muscle response. This approach relies on having numerous representative annotations available. You might recognize the problem here. An algorithm that is based on an input from a single researcher or a research lab will end up great at mimicking that researcher specifically. It might not be generalizable to the style of the other researchers. Clicker can aid in this aspect. We provide a vast dataset of annotated TMS-EMG responses from a variety of researchers that can be used for algorithm development. Furthermore, our data are not annotated by a single person, but we provide multiple annotations from different researchers per each response. This vastly reduces the amount of researcher variability influencing the algorithms. Our aim in the future is to have a dataset where each response features a distribution of annotations, which can be then used to estimate the reliability of the data. This researcher greatly benefits from having this large annotated database available. Next, I will talk about how to gather these data. Our final hypothetical researcher is a group leader whose research group has been conducting TMS-EMG experiments for years. Each passing graduate student and a postdoc have accumulated their own contribution to the knowledge they have on the topic. Their findings have been published in numerous scientific articles. These data, which the publications are based on, are stored on various media. Some are on laboratory servers, others are stored on memory sticks and personal computers. Altogether, there could be tens of thousands of annotated responses that form the basis of their knowledge they gathered. What are these data good for anymore? Probably not much, to be honest. It is unlikely that they will be reanalyzed or used in another publication once the researcher that conducted the experiments has moved on. This does not have to be the case though. These data would be extremely valuable to a researcher who is analyzing brain stimulation data and also to those who are developing algorithms. The clicker provides a way to make use of existing data that would otherwise be left unused. Data that has been published is rarely useful to keep in private. There are numerous researchers out there who are looking for this kind of data. Our hypothetical group leader realizes that there is no downside for publishing these data. The person advises their researchers to contribute all published data to the clicker database so they become available for the community to take advantage. Now, the thing many of you will have in your mind is, this is highly sensitive biometric data, it cannot be made public. This is a sensible and good approach to data safety. We of course have put a lot of thought into this matter. We have come up with a few ground rules that should make using the database safe. First of all, we only store anonymous data. Now what this means is that when the data are uploaded to the platform, all metadata, including any dates and identifiers, are stripped and destroyed permanently. This is done on the computer of the contributor. The original data are not transmitted to the servers. At this point, we provide the contributor with an ID they can use to track each part of the data if they wish to do so. These IDs do not leave the local computer. The user can manually review the data before it's sent to the clicker database. On the server, only raw data will be stored. Furthermore, we only store the data contributor, the accepted license, and the date of upload. Notably, we cannot retrieve any more information than that even if the contributor asks. Only possible links to the original data are left with the contributor themselves. Secondly, the data are split into standard size epochs and resampled to a fixed format. This breaks the connection of byte-to-byte comparisons between the original data and the stored data. As an additional measure, we can, for example, add a very slight amount of white noise to the data so that even comparisons between curve shapes becomes more difficult. However, we must take care not to do anything to the data that might affect the analysis results later. No one can unfortunately guarantee 100% data safety. However, let us consider two possible scenarios. If someone has full access to the clicker databases, they can find the following information: the epoch data, the annotations given by the users, and the collections which the data belongs to. Notice that none of these data are still private. The collection states of the user who contributed the data, the license they chose, upload date, and possibly the user informations they have given. Even though having user data leak out of any system is bad, none of these are directly compromising the security of the data of any of the subjects in the original experiments. On the other hand, if someone gets access to the original data and would like to know if the subject they are looking at are represented in the clicker database, they would first need to transform the data using the same tools as the original author, using the same keys. Then they would need to heuristically compare the epoch waveforms to each waveform in the database. While this can theoretically be done, it is computationally intensive, doesn't guarantee accuracy, and it requires access to both original data and all of the clicker database. My aim in this presentation has been to show you that we have developed a system that provides a unique opportunity for the TMS-EMG community to support each other, provide new opportunities for research, and advance the science in this field. Currently, we have a working prototype of the system running, which you can go and take a look. The link is in the last slide. We have a small example dataset running, and we are currently revising all of the front-end software. Unfortunately, the new interface was not ready for this presentation, but if you check in a few days, it should be available. I suggest that you go and take a look at the system online and consider contributing to the analysis of the database. If you'd like to contribute to the annotation of the data, you can go to the platform and start clicking. However, if you would also like to receive acknowledgement for your work, you need to create a user account to the system so we can track your progress. The user interface is still a work in progress and we'd appreciate any feedback you can provide. If you're a researcher or group leader with TMS-EMG data and you would like to contribute, please contact me directly. You can find my contact information from the URL shown on the screen. Thank you for listening about the clicker system for annotating TMS-EMG data. I would like to acknowledge funding from the Janne and Aatos Erkka Foundation, as well as from the European Research Council. Finally, I thank all the people who have contributed to this project in one way or another.
Thank you for your interest in Rogue Research and our products for non-invasive brain stimulation. We offer the BrainSight neuronavigation system for transcranial magnetic stimulation and focused ultrasound placement. We also are releasing our Elevate TMS, controllable TMS device. We also offer functional near-infrared spectroscopy with BrainSight NIRS. BrainSight TMS is a complete neuronavigation system for TMS coil placement and focused ultrasound. It includes an integrated 2-channel EMG. We also offer a flexible subject chair and coil holder. The system supports multimodality acquisition, so we can easily combine TMS with EEG or TMS with FNIRS. The system also integrates with the TMS robot. The navigation software supports both subject-specific MRI or the ICBM 152 average brain template. More recently, we've released a version of BrainSight that interfaces with the SID-NIBS modeling platform for more sophisticated and easy-to-use current modeling. When performing your TMS session, you can use our intuitive display, which includes a 3-dimensional representation of the coil, the subject's head, and the brain, or our easy-to-interpret bullseye display. Our integrated EMG allows us to record the EMG responses from the TMS pulses, as well as the position and orientation of the coil at the time of the pulse. This allows us to map the EMG data back to specific locations in the brain and generate these useful heat maps of EMG response. Our subject chair is very flexible. It works equally well in reclined or upright positions. It is height adjustable and rotates. It also includes unique TMS coil holder arms. These arms are very flexible and very intuitive in placing the coil over the head. The system also supports multimodality acquisitions. We can map the locations of EEG electrodes in common montages onto subject-specific MRI. You can then use the navigation system to ensure correct cap placement or record the locations of each electrode for export into text files that are commonly used in EEG analysis software. We are proud to release our new Elevate TMS device, which is a controllable or CTMS device. It is the most powerful and flexible TMS device available. It gives us control over pulse parameters that are not available in other devices. These include the pulse width as well as the directionality. It supports also biphasic and monophasic pulses. It includes a modern user interface using a large touchscreen. We've also put some thought into the coil design with removable handles that allows us to use different types of handles for different applications, as well as direct connection to our holder arms. BrainSight NIRS allows us to use functional near-infrared spectroscopy to record brain activity. Our design is very modular. We created these 8-channel modules, which include 8 high-sensitivity detectors, 4 dual or 6 dual wavelength sources, which can be also reconfigured to be 4 3-wavelength sources. We also include 4 proximity detectors. These detectors are designed specifically to record close to each source to measure the predominant physiology signal, which is thought to confound NIRS data. The NIRS enclosure can accommodate 4 modules, so we can configure a system to go from 8 to 32 high sensitivity detectors, 4 to 24 sources, or 4 to 16 proximity detectors. The low-profile optodes can be easily combined with EEG to do combined EEG and NIRS acquisitions. The low profile and TMS compatibility also means that we can easily place the coil directly over the optodes with a small distance penalty. Our caps offer very flexible montages. We can create montages for very specific brain regions, and using the navigation software, easily visualize where in the brain a proposed montage will be recording from. Using the navigator, we can then ensure the cap is correctly placed, as we would also for an EEG cap. We can also digitize the optode locations, and using the navigation system for analysis, we can then map the NIRS signal back to the region of the brain that we were recording from for better visualization and interpretation. More information can be found at www.rogue-research.com or send us an email at info@rogue-research.com. Thank you for your time.